All right, so it's 4 o'clock. Daniel, are you there?
I'm here, yeah. I think there was just an issue with some new headphones I had, so I'm just going back to old school, just holding my telephone.
No worries. We have had plenty of technical issues on our end as well. All right, so welcome everybody. This is the Longevity Biotech show here on Clubhouse. I'm your host, Robert, along with Nathan Cheng. And today we are interviewing Daniel Ives of Shift Bioscience. This show is where we interview scientists, entrepreneurs, thought leaders, advancing the state of the art in longevity biotechnology and how this will impact us and the world in the decades directly ahead. What we're going to do is we're going to have a few minutes of introductions, about 25 minutes of prepared questions, then about 25 minutes of audience questions, and a few minutes of lightning round at the end. And then there's an open-ended chat for as long as we can go for anyone interested who has the time. So if you can just hold your questions until the half-hour mark, that would be great. Also, a reminder to everybody that we are recording this. So as per the Clubhouse rules, if we have you on stage or if you raise your hand and you agree to come on stage, you're consenting to our using your voice and image in the recording. All right, with that, maybe we can start with the intros. Nathan.
Yeah, sure. Thanks, Robert. So my name's Nathan. I'm the founder of longevitymarketcap.com, and there's an associated newsletter there that you can check out. It's once a week, just covers different developments in the longevity biotech industry. I'm also the founder of longevitylist.com, so it's a website where you can find jobs, companies, and investors in the longevity industry space. So, yeah, Robert?
Great. Yeah, I'm Robert. I've worked in biotech in bioinformatics and research software engineering capacity for about 10 years. And last year, I've gotten more involved with the aging biology research field and have been trying to engage in the longevity biotechnology venture space as well. So for now, co-hosting this show and working on a few other projects on the side. Daniel.
Sure, yeah. I'm Daniel Ives. I'm the founder and CEO of Shift Bioscience. And when we set up, we were solely focused on the mitochondria, largely inspired by Aubrey, who basically said mitochondria are one of the most important things to focus on. We've got a new focus now, which I can sort of touch upon perhaps a bit later. But it's really sort of driven by epigenetic clocks and that technology allowing us to audit the different hypotheses, like is mitochondria still the most important thing, or is there something bigger than this? So yeah, that's where I'll leave it.
Great. Well, the focus of the call is on you today. So, and I know that Nathan invited you on, and I guess it's more familiar actually with everything that you're doing. So I'm going to pass it over to Nathan to lead the prepared Q&A here.
Great. Thanks, Robert. So as I said earlier, the sort of purpose of this talk is to discuss Daniel's history and his experience actually bootstrapping Shift Bioscience, his company, from the early days to where it is now. And the idea here is that we want to be able to inspire others who are interested in potentially founding a longevity biotech company to follow in his footsteps or just learn from his experience so that they have maybe like a roadmap or a template of how to even get started. So with that, maybe we can just start off with Daniel telling us about where your story begins.
Sure, thanks, Nathan. So basically, it all begins in 2008. I'd done a biochemistry degree in Imperial College in London, and I just really enjoyed everything about biology, the chemistry. Put those together, it's biochemistry. But in 2008, the world was in a bit of a state with the financial crisis. So I was looking to go.
Looks like we lost him. Okay. Technical issues. There we are. Okay, back. He's back.
Sorry, guys.
No worries.
Sorry.
No worries.
I promise this isn't deliberate. Yeah. So in
No.
2008, there was a financial crisis, and I've been looking at sort of farmer roles, but they'd all dried up. So basically, it was, do I take something that's a dead end, or do I basically do what's the coolest thing I can do with my skills? Basically, not very responsible decision-making process. But at the very same time, Aubrey had released his book called Ending Aging, and it's just a fantastic book. I don't know whether that's the first book someone would read now, but at the time it was just such an eye-opener. It just sort of made the idea of getting hold of aging and reversing it a reality. And I bought into this. I just thought it was fantastic, and I basically just searched for research projects that were basically along the lines of Aubrey's framework, the SENS framework, different types of damage. And Aubrey sort of listed mitochondria at the very top, and so I actually found a research project in Cambridge, UK, that was a joint project between Aubrey and an individual called Ian Holt. And I applied to this project. The project was basically to try and take the mitochondrial genome and put it into the nucleus. So for those not familiar, the mitochondria is a little organelle in the cell, has its own vestigial genome. Most of its genome's transferred to the nucleus, but you've got this tiny genome left. And the idea was, let's just transfer the rest of it, right? Let's finish what evolution started. And that was a very compelling project. Unfortunately, that project being discontinued in Cambridge, so we just encountered technical difficulties. But there was a side project in the same lab that was trying to achieve the same thing, which was to get rid of mitochondrial DNA mutations. Basically, these mutations basically take over cells, cause lots of problems. So you can get over that by transferring it to the nucleus, and then it's safeguarded, or you can try and persuade the cell to get rid of bad genomes and basically enrich good genomes. It's something called bias segregation, very obscure phenomenon. But that's where it basically started. So, I mean, the idea was, I've got some skills, I'm a smart, I thought I was a smart person, but I won't be the judge of that. Should I just go for it? Like, bet on yourself. And so I decided to, and that was a step one.
Great, yeah. So starting from that project, looking at the mitochondria, how did you proceed to come up with an idea for your startup, Shift Bioscience?
Sure. So I'm going to talk about quite a niche topic here, but it was sort of overwhelming to me, right? The significance. So people that are interested in something called mitochondrial heteroplasmy, it's either all of their life, or other people just don't know anything about it. So basically, there's these individuals with a disease. It's called MELAS. It's a rare mitochondrial disease, and what happens is you just inherit your mitochondria from your mum. So it's just monoparental inheritance. You don't inherit 50% of your mitochondrial genes from your dad and 50% from your mum. It's 100% from your mum. So you could sort of say that you're running your mum's energy system. So these individuals with MELAS, they inherit a mixture of mitochondria, and basically there's a subpopulation in the cell that contains a wild-type genome, and there's a subpopulation that contains a mutant genome that causes a bunch of pathologies, neuromuscular issues, basically high energy tissues that you need a lot of energy, need a lot of mitochondria, and therefore you get symptoms. So what my supervisor, PhD supervisor Ian Holt had discovered is that if you take one of these patient cells that basically contains two sort of teams of mitochondria, the good team and the bad team, and you pop out the nucleus from the patient and you stick in the nucleus from a cancer cell line, a particular cancer cell line called A549, that nucleus can basically get rid of the bad team. So we don't know whether it's preferential degradation of the bad team or it's selective reproduction of the good team. Like we're not able to discriminate at the moment. But basically this A549 cancer cell line can get rid of the bad team, all the way to 100% good team, which is 100% wild type mitochondrial DNA. And interestingly, if you have the nucleus from a different cell line, I think it's called 143B, it's an osteosarcoma. You stick that in the cell instead of the A549 nucleus, you get the opposite phenomenon. So you basically, the bad team takes over with time. And there's other cell lines that do this as well. So there's A549 that does something good, which is like, basically get rid of the bad guys. And then there's all the other cell lines that do something bad, which is the bad guys win. And this was like the 90s, right, when this was discovered. And I showed up in 2009. And what had changed since then was that there was a big database called NCBI-GEO. And it contained, just loads of data sets for A549, transcriptome data sets. And there was loads of data sets for the other cell lines, that do the opposite. And, my bet was that I could look at those transcriptomes, compare them bioinformatically, and distill what is it that's so special about this A549 cell line that allows it to do this magic, which nobody knew what was going on. So basically my PhD supervisor, Ian Holt, he said, well, it's not going to cost us anything, so why not? Yeah. So, because there was relatively little risk to him, apart from my time, of course, he let me do it. And I was just so happy to work on this, because that's why, I was really there to basically just try and find technologies to overcome problems with the mitochondrial genome. A lot of other PhD students in the lab, they get assigned to a protein. So there was a protein called NIPSNAT1. There was another protein called what was it? What was it? Anyway, it doesn't matter. Just different proteins. And I'd watch some of these PhD students, and it's really pot luck. Like, you get assigned to a protein, and at the other end, you might have something very interesting and a great PhD to write up. But you might get assigned to a protein that's basically a dud, and you just have to sort of, damage limitation at the end of the PhD. And I didn't want to, take the risk with my time. I really wanted to just, do something that would, had the highest potential to work, even if it was risky. And so I basically convinced Ian, let me do this. I risk my own time, but it doesn't cost the lab anything. And the short story is, we were able to figure out what this A549 cell line was doing. It's not actually a mitochondrial pathway. It was a pathway outside the mitochondria. It's a stress response pathway. But yeah, it was so difficult getting to it, because it was on me to prove what was happening. So I did all this computer work, and then basically just so much cell culture. People used to joke, like, do you get any light? Like, you're so pale. I've got quite a pale complexion anyway, so I do need a bit of sun just to look human. But I wasn't getting any sun. I was just like, I had hundreds of cell lines in parallel going on, for ages, months and months. And occasionally, at the end of a 3-month time course, I get contamination. I'd have to start all over again, so which is quite frustrating. But it was worth it in the end. That's the good news, right? It was worth it in the end. So we managed to find this activity. And I guess the moment I knew this activity could be distilled for MELAS, which was a rare disease, for me, it was always about the bigger picture. Yeah, it was, okay, this is a tool for aging. And if the lab's not going that direction, this is sort of a natural fork point. And, my supervisor was interested in aging, of course, but academic labs, they do have to focus, they do have to publish. And if you start doing, sort of, you go out into the wilderness on a vision quest, and, you don't bring anything back, that's academic fuel that's been lost, right? That could have been spent creating some sort of story about something. Yeah.
Okay, so that's great. So you were doing your PhD, learning about all this interesting stuff in mitochondria and heteroplasmy. At what point did you come to the realization that you wanted to start a company?
So I didn't come to the realization immediately. So it was, I tried to do it within academia first. Yeah. So I was working at the MRC, Medical Research Council, which is a major funder of life science in the UK. And basically you can talk to them about licensing IP, and you can try and get into those discussions, but it was all very difficult. And then they have a very strange policy, which is if you have anything valuable, the MRC get 90% of the upside, right? So if you start a company and you do anything with that company, the MRC basically will have 90%. And it felt more like a deterrent rather than a commercialization program. It was more like, let's stop academics going off and doing distracting things in the real world. Like we need to keep them sort of doing the academic groundwork. So like I tried to go that direction and raise some interest and try and bring Ian along with me. But it just got slower and slower. Things slowed down. We were just waiting for emails from people, and they'd never arrive. And I just thought, okay, this can't be the only way, right? If the project's good, which I believed in, I've just got to change my environment, or I've got to at least try and find a new environment for this project to keep it moving forwards. So that's when I just basically started reaching out to people that knew better as far as drug development's concerned. There was a great guy called Simon Westbrook that I was introduced to. So he used to work at Pfizer. And then Pfizer used to have a headquarters in Kent in UK, global headquarters. And when they shut that, the facility remained. And Simon had basically been working there a long time. And he took over some of that facility and set up a company basically with his own IP that he generated. He knows a lot about just big pharma, commercializing things, what people want to see, what's not so important. And I basically started talking to him more and more. He sort of gave me an idea about what I should be doing, what I shouldn't be doing. And then the question was, let's go out and find somebody that's basically as interested as we are, and in doing something about not necessarily aging, but look, we've got a technology here and we want to move it forwards, and do you want to back us? And so I mean, how much do you want me to keep going, Nathan?
Yeah, keep going. Just want to get to the point where you actually started the company, and how you actually got to a point where you could develop enough science to pitch it to investors. I guess that's where we're heading.
Sure. Yeah. So I was sort of reaching out at the time, trying to get it done in academia, not getting very far. And sort of what happened was my contract was cut short. So I was a postdoc at the MRC. My contract was cut short by a month, and that obviously that's not great, because you were expecting it. But the MRC have a great policy, which is if you basically get made redundant, you get this quite generous redundancy fee. So I think in my case it was about £10,000 or $15,000. And I was just itching, right, to just go out and go in my own direction. And I was getting lots of no's and no's, within the environment that I occupied. So the redundancy, I think Ian asked me to stay on. I just said, look, Ian, I really want to do this, and it's slightly different. And for me, this was a natural break point. So basically, I didn't sort of stick with the academic lab. I just said, look, I'm going to try and do something. And I basically just spent that money on experiments. And you might ask, okay, how do you just do experiments, right? You can't just do them in your house on your breakfast table. But what many people might not know is that there's plenty of contract research organizations out there. So these are basically pay to access labs. So there's scientists in these labs. They have all the equipment that you'd have in an academic institution. And they take experiments from lots of different directions. So, you can basically do an experiment or conduct an experiment very cheaply, because you just pay your materials and your fraction of the overhead for that contract research laboratory. So what we were able to do, we did a bit of work basically trying to find, we discovered a very rudimentary or crude technology that had real toxicity issues. So we basically firstly tried to do a bit of work. But how do we find something that's therapeutically got a better chance, right, of getting from start to finish? So what is the potential target, right? That's the first question. Like, where is this molecule binding? Because we didn't know that. We just knew there was a pathway activated, and this molecule activated the pathway. So where's the target? And we basically found what we thought was a potential target. We screened a bunch of molecules that were similar to the original molecule, just modified in different ways, structurally similar but different. And we found something that was much better at binding the target that we thought was the target, putative target, yeah, not confirmed. And it so happened this molecule was way better as far as toxicity is concerned, so much lower toxicity. Yeah, we did this all in a contract research lab, and I think I basically spent that redundancy money, and we generated some really interesting data. So at that point, again, I didn't know whether this was going to be a company or not. I just wanted to go in my direction, right? It was like, there's a goal, which is, let's make a big impact on this process. How do I get there, right? So I had some money, I spent that, but then I ran out of that money. So I basically asked friends and family at that stage. So I think my dad put in about $40,000. I didn't want to ask for any more, because you do have to share Christmas with your relatives, and if you've lost $40,000, it changes the dynamic, and Christmas doesn't have that warm feeling anymore. So you're risking that. So I sort of put a cap on what I'm going to take from my dad. And then I think I thought my uncle put in a bit of money as well. Obviously not too much again, because it could all go south, especially with biotech, and especially with something that's a big unknown like aging. And then finally, I asked my girlfriend at the time, can I use your savings to do the next experiment? And she said no, which was the right answer, because she'd worked hard to build up that money. And she said you should really go to an investor, like somebody that can afford to lose it, which was very good advice. And then we basically, and I'll keep going. Sorry, this is quite a long monologue. I'm fortunate that in my family, I have an entrepreneur. So my dad, there's interesting stories here, but basically in the mid 80s, late 90s, he set up a company which was it was like a precursor to the internet. It was local area networking within buildings, right? Not sort of between buildings and between everyone in the world, but it was local area networking, and this generated a lot of buzz in Cambridge at the time. So I think at the age of 25, he had 200 people under his employment. He was approached by Microsoft, and there was a whole sort of fallout with his co-founders, because Microsoft wanted to acquire. He really wanted to go ahead, but then they said no, they're going to take on Microsoft. Anyway, there's this whole excitement, and he basically had to resign from that company. If the acquisition had gone through, he would have had early Microsoft shares, and I don't want to say how many billions, but it's just ridiculous. So he has all this experience, and he had some great contacts in Cambridge just from ages ago. And so he did biochemistry as a degree, and I kept him in the loop with what was going on. He just naturally got interested, and he said, well, let's go back to my network, which is a bit out of date, but let's go talk to some people, right? And if it's interesting, that's great. If it's not, we'll find out sooner rather than later. And so we went to talk to a guy called Herman Hauser. So Herman set up a company called ARM. So ARM is they design most of, I think, an ARM microchips can be found in most mobile phones. Basically, Herman Hauser runs a firm in Cambridge, UK called Amadeus Capital. And so we went to talk to Herman, sort of basically, this is what we're doing. Are you interested? And it was slightly out of his area. Yeah, he wasn't a life science guy. So, well, he does do life science stuff now, but it's not his specialty. And he was interested, but I think he was polite, and he just said, look, this is very early. This is basically an experimental project. It's like a science project, right? It's not like a company, which was correct, by the way. And he said, why don't you talk to my friend Jonathan? So that he had a friend called Jonathan Milner. Jonathan's had set up a company called Abcam. I can't remember when it was, but Abcam had been quite a success story in the UK. So like multi-billion dollar, multi-billion pound company. Sorry, correct currency. It's a life science or reagents company, and Jonathan had basically done very well out of that and become a seed investor. And so we went to talk to Jonathan, and Jonathan said the same thing. So he was like, yeah, this is very interesting, but very early, not a company at the moment, but keep me in the loop, right? So keep me in the loop. And so we actually sort of went back, dug a bit deeper with our own money, and did some more experiments and actually moved forwards again, right? We kept making progress against the odds. I went back to Jonathan, and Jonathan, I think, was just impressed. He was like, well, I can't believe you did anything with the money that you spent. And he basically said, look, I will gift you some money. Because I didn't know if this was going to be a company. It could, we were thinking like maybe non-profit, maybe company. He says, I'll gift you a certain amount of money, just a small amount. Condition being, I get first refusal if this does become a company. Yeah, so that was the deal. And we used the money, we spent it well, like we generated some good results. Basically, we showed that we found heteroplasmy, so basically a mixture of bad mitochondria, good mitochondria in idiopathic Parkinson's disease cells. So it's actually a novel finding. It was, there's reasons why we found it and others didn't based on technology. But we found that there was a mixture, and we showed that with our newfound molecule, we could basically get rid of the bad mitochondria in these Parkinson cells. We showed this to Jonathan. He was like, fantastic. Let's do it. Yeah, let's start this company. And I think, Jonathan didn't know about aging at the time. So I sort of introduced him to, the idea that aging is something that can be overcome. It's not this pipe dream. It doesn't require you to suspend belief. It's actually experimentally tractable. And we've got great, there's a great opportunity that I'm working on. There's plenty of things out there. And Jonathan very quickly, because he was a scientist himself, right? He was a scientist turned entrepreneur. He quite quickly basically, started looking this up and, doing some digging, got really excited, like very, very excited, like shouting from the rooftops, excited. And he was basically our champion and cheerleader from that point onwards. But that's how we got our first investment. And basically Jonathan's been there from the very beginning, and he's continued to support us from this day. And I think basically without Jonathan, we wouldn't be having this conversation.
Well, yeah, that's an amazing story. So how much do you think you ended up putting into the company before you were able to get outside investment? So how much money did you spend on like the CRO stuff?
So I'll try and do the conversion to dollars in my head. So I think personally I put in about $40,000 myself. I didn't mention I'd done an internship at an investment bank in my second year of uni. And investment banks, before the financial crisis, used to pay stupid money even for internships. So I had like $15,000 just from 3 months of work. It was like ridiculous. I didn't go into that industry, but I had some money left over from that, so that was part of it, and then my redundancy, and then just some other savings as well. And then I think my dad put in an equal amount. I think Jonathan put in I won't say, I don't know whether he'd want me to say or not. He put in a small amount, roughly equivalent. I had some really kind people that just heard about what I was doing and just donated. So like occasionally I'll get $1,500. It's amazing if you just talk to people, right? Especially people that are sort of well off. You can just talk to somebody, and if you can share your excitement with them effectively, all of a sudden, you've got a road forwards. It's complete opposite to academia, right, where everything was slowing down. I talked to people, and it was like, wow, now I can do this. Wow, I can do that. It was sort of a revelation. What the overall number would probably be about $70,000, $80,000. So it's not negligible, yeah, but it's certainly something to think about, yeah. If you've got, if you're trying to, I don't know, if you're trying to balance the risk, yeah, that might be a bit too much to stomach. But I think for me, I guess I just wanted to go for it, and I was young. I probably watched a few too many of those YouTube videos, like motivational speeches and things. So it was about $70,000 or $80,000.
Okay, that's obviously not everybody has that kind of money in their pocket or whatever, but in terms of biotech, I think that's very, very reasonable. Because I asked Celine Haliua of Loyal. She's the founder of Loyal, another longevity biotech company. Asked her how much would it cost for them to get to sort of an investable amount or an investable, sort of company that would be like get enough data for investors to be able to invest in her company, and she said that they spent maybe like $500,000. So this was all on like a Twitter thread. So yours is very reasonable, like in terms of biotech, I feel like.
Yeah, just a couple of points, Nathan. Firstly, in the UK, we're very cheap. Yeah, so we we're very value focused. It's like, oh, why not take an extra risk and save 50%? Yeah, which you wouldn't do for a helicopter ride, for instance. So that's the first thing. The second thing is Jonathan. I think we were very lucky with Jonathan. So, when we went to see him for the first time, he's got nature journals like just on a table to the side. Like he's really passionate about science. So for us to basically open up a new chapter in the scientific story, or open up a new page that he wasn't aware of, it was just fascinating for him, right? It was like a new area. And I think there was a lot of good timing. Maybe it was a year later, it might not have happened. Maybe it was a year earlier, it might not have happened. But it was a good combination. So maybe that $80,000 was a moment in time. But I don't think it would be so dissimilar now, especially with the interest in longevity. I think you could do simple things and get quite far.
Yeah, great. So I guess the next question would be, maybe you could talk to us about the patent filing process, because this is sort of important for early stage biotech companies. They're trying to raise money, but they also want to like have some sort of protection of IP. Maybe you can tell us how you went about doing that.
Yeah, so I basically dived in at the deep end, jumped in the deep end, sorry, not really knowing what I was doing. I just thought, okay, I should file a US patent, just because that would be more valuable, and from my naive perspective, and I just sort of read some online articles about what you should put in a patent, how to file claims, things like that. But actually, and then I filed the patent. I think there's a US PTO patent very early. But the problem is you don't know what you don't know, and if basically knowing what I know now, you should do some basic checks. So you should just do a check of what's out there, what's the state of the art, right? So what's obvious, what's not obvious. If there's things out there that overlap with what you're doing, you need to sort of just consider, maybe narrow the claims of your patent a bit. So you just narrow it down to something specific. And yeah, basically I learned the hard way just by wasting my own money, and how to sort of do patents properly. As far as what the patent is for is concerned, a patent is, obviously it's you can protect your intellectual property and make sure somebody isn't ripping you off. But by the time you're getting ripped off, you must be doing pretty well. Yeah, in biotech you must have got to quite a significant endpoint. So actually, in the early stages, a patent is more about creating a question mark for somebody else. So if somebody else is doing something, you sort of blocking their path. It sounds a little bit counterproductive, right, as far as the overall field is concerned, but you're just raising doubts, that somebody could pass through this intellectual property space, and not have to pay you something, whether that's immediately or whether you litigate later. Yeah, so you just sort of, you just need to plant that flag early, and then somebody has got to just try and sort of balance the risk of you doing something. So that's one use. And then the second use is just, from an investment perspective, if you haven't protected what you have, and somebody gives you a load of money, well, as far as they're concerned, that money is just going to leak out, as fast as it got in. So you just need some basic sort of protection. And obviously with the money, you can sort of revise your patent and make it a bit more watertight. It's like, building your moat. But that's what I would say about patents. And I think, where we are now, by the way, there's really efficient strategies. So as long as things don't make, publications, they don't become common knowledge, you can do something called withdraw and refile. So you basically file, and if nothing's happened in the world with respect to what you want to do, in sort of a year, instead of progressing the patent, which gets more expensive and expensive, you can just quietly withdraw and refile, and you've still got the flag is, you planted the flag first, but nobody basically sees any of this, and nobody can see the patent in this first year as well. It's a very efficient way of doing things and keeping your patent fees down, but also keeping your patent life up, so you're not losing patent life in the process. So this is something I'd recommend, but you're taking a risk, yeah. And like I said, we have this tendency, I'd say, in my experience, we take more risks here that in the long run might undermine us, but we can do things more cheaply.
Okay, great. Yeah. So where we left off, you finally were able to get outside investment from Jonathan Milner. And then what happened next? And how did you get from that point to where you are now? What is your company?
Sure. Yeah. So we got the investment from Jonathan. Obviously, when you get your first investment, it's hugely exciting time, right? I think it's like I think there's an Elon Musk talk, and he talks about this. It's like peak optimism, peak happiness. You've got all the money in the bank, right? It's just it's like, how do I make sure I don't spend this too quickly and waste it? It's almost like this exuberance. And then you just need to get on with the business of actually trying to do something and move it forwards. And that's obviously very difficult. So, we started doing experiments, and I think this was 2018. And so I wasn't, I was a mitochondrial biologist. I was a little bit, I was blinkered, right? I was guilty of being blinkered. I knew a lot about mitochondria, but I didn't know a lot about outside of mitochondria. So as far as I was concerned, I'd read Aubrey's book. I'd sort of, enriched my knowledge of mitochondria subsequent to that in my PhD. Everything seemed to be explained by mitochondria. It was a perfect little world. But during the process of sort of pushing Shift forwards, it just occurs to you, because you get these questions and people sort of will test you. They'll be like, well, there's this other thing that seems to slow down aging, and it hasn't got anything to do with mitochondria. And you get into this dynamic where you're trying to say, I'm the best and these people are the worst, and it becomes a competition, and that's not the correct way to do things. The correct way to do things is to try and continually question your assumptions, like basically audit your hypothesis. So my hypothesis was mitochondrial DNA mutations create bad mitochondria, and that drives aging, and there's evidence to support that. And we've basically gone ahead after the Parkinson's cell experiments. We'd taken a mouse called the mutator mouse. So this mouse is a premature aging model, and the aging is caused by an increased level of mitochondrial DNA mutations. It doesn't mean those mutations cause aging in you and me, but it is provocative, right? The fact that you turn up these mutations, it creates aging in the mice. And I think it was like a Nature front page. It was like aging solved or something like that. Yeah, it's just totally out there. So we basically put our drug in this mouse, and we got some really nice results. The mice visibly, they look younger, so we slowed down their aging phenotypically on the outside. And then whilst I think we were halfway through this study, and then we became aware of the epigenetic aging clock. So I'm sure most of you on the call are familiar, but for those that aren't, Steve Horvath discovered a robust biomarker of aging in 2013 in humans to begin with, actually, not in a model organism, in humans. And the accuracy is phenomenal for this biomarker. Like, it's good for a biomarker, let alone an aging biomarker. And subsequent to that, Wolf Reich, who's at the Baber Institute in Cambridge, defined a mouse epigenetic aging clock. And for me, this was a perfect opportunity to test my assumptions, to test the idea that mitochondria is the best thing to be targeting as far as intervening in aging is concerned. So we basically embedded one of our employees in the lab of Wolf Reich's lab at the Baber Institute. So there was no service at the time, right? You couldn't do these measurements without the academics. That's how involved it was. So we actually put one of our own people into the lab to get the experiments done, because the lab couldn't help us. And we were able to show that we could slow down the mouse epigenetic aging clock by 50%, which was a lot. I think rapamycin in a different model, it wasn't a progeroid model, had slowed it by 27%. So 50% was a lot. So we were like, oh wow, this is great. We've got something here. But then all of these other waves started crashing, like positive waves, right? So you had epigenetic reprogramming, where you weren't just slowing down aging. You were doing much more than that. You were reversing aging. And it was very comprehensive, right? The clock was, going from 60 years to 0 in 20 days. All of the other obvious hallmarks of aging were reversed. This is sort of a fantastic technology. And I remember I was at the conference Undoing Aging in Berlin. I was there to talk about mitochondria, and then I think it's with Vittorio Sebastiano was there to talk about Turn Bio and epigenetic reprogramming. I just remember watching his talk, and it was fantastic, right? This epigenetic reprogramming, and the fact you could reverse, it was just such a watershed moment. The fact you could turn things back, not just prolong the inevitable. And he was presenting, and one part of me was like, this is amazing, right? This is fantastic, almost my inner scientist, my personal interest. But then on the shift side, it was like a dagger to the heart. It was like, oh my God, this is just totally obsoleted, my approach with respect to, the summit, which is human intervention in aging. So there's an important choice you have to make at that stage, which is, do I continue down the path that I'm on, because that's where I've invested time and money, investors' money, or do I try to adapt? Do I try to sort of steer what I'm doing in a different direction? Because ultimately we just we want to be working on the best stuff, not just what we worked on before. We shouldn't be defined by legacy. So we got interested in the epigenetic reprogramming side. And in particular the clocks. So the clocks, I don't think this is commonplace to frame it in this way, but the clocks are very useful in two perspectives. So firstly, they're a nice readout. So if you're mucking around with some biology in the cell, you've got an instant readout, or near instant readout of aging. You don't have to do a lifespan analysis, so you can play with the Golgi apparatus. What does the clock say? Let's play with the endoplasmic reticulum. What does the clock say? You can get a nice, question and answer response like quickly. So that's one thing that clocks do really well. It's like a dial, you can sort of, check what's going on. But the second thing, which is actually more interesting, is the clock is a window into aging biology itself. So, when Steve Horvath came up with the original clock, it was 353 CPGs. And an obvious question was, well, what are those CPGs? So, what's special about them? Which genes are they near? And, Steve does actually explore this in his 2013 paper. But the short answer is it's very difficult to make a biological inference from CPGs. But just because CPGs are sort of very distant from the functional level, first of all. Secondly, Steve's clock, if you remove those 353 clock sites, just put them to one side and basically run the algorithm again, the clock training algorithm, you could basically train an equally accurate clock. So there's this huge degeneracy, right? So it's like there's regularities in epigenetics that aren't confined to those 353 sites. So you can basically just find this regularity popping up elsewhere. But the principle, that you could, the clock is a window into aging, is actually very exciting and valid. And so more recently, I think he's created, was it the mammalian clock? So this is a conserved clock, every tissue, every mammalian species. And the identity of those CPGs is actually, that you can make a better biological inference. And so, I really got interested in this, and we had an intern join us at Shift that was really interested in this. We were initially interested in single cell clocks, trying to, find out what's going on below the level of the population. So, what's going on in single cells, the different trajectories. But we arrived at, the clock as a window into aging biology. And that's sort of where we are now. Now, we might find out that mitochondria are still at the center of the universe, right? It's just, you need epigenetic reprogramming to do something to the mitochondria, and then they take over. But we might find out it's something completely different. And we also might find out that there's completely new sort of frontiers of aging biology, which is exciting as well. And either outcome, we should do the experiment, because if it is mitochondria, we've got way more confidence as far as putting money behind that organelle is concerned. If it isn't mitochondria, we can, basically change course and move forwards in a more productive direction. What I don't want to be doing at any stage is flogging a dead horse. For me, it's the summit, the Everest is human therapeutic intervention in aging and whatever is the most effective. If I find myself deviating from that, I need to ask some hard questions of myself, and it's about staying faithful to that. And it's very easy to become unfaithful and just say, oh, this has been so much effort, I'll just settle for less. But I've tried not to forget what the real goal is.
Yeah, that's awesome. Yes, staying true to your North Star of your mission to treat aging as a disease. So I guess maybe one last question before we open it up to the audience. I know we're running a little late. Maybe you can just give some advice, some general advice to potential founders. You having gone through this process of starting a company with your own money, bootstrapping. If someone else wants to do something similar to what you have done, what would you say they should avoid? What things should they try and stick?
Yeah, so what I would say first of all is that Shift Bioscience didn't just happen. I didn't just have the idea, let's start a longevity biotech company. It was more organic. So it was like, look, do I have confidence in myself first of all, and then secondly, are there technologies out there that I want to work on and push forwards? And the home for those to begin with was actually an academic institution. It wasn't a company. That situation might have changed now, but at the time it was, what's the best environment to progress this? And then there's a lot of personal questions here. It's like, how much risk am I willing to take? How much am I willing to sacrifice? When I was trying to do these cell culture experiments, people going on holidays, people buying houses. I was just renting a flat on the basically as close to my institute as possible. You have to decide how important. And I think something I haven't covered was I almost gave up multiple times, right? There was many points where I was like, is it worth it? It's like, everything I've done worth it. And the longer you leave it, right, the more you continue down this path, and then it doesn't work out in the end, the more time you've wasted. So there's just a few times where I was like, look, if I don't get this result, I'm walking away. Like I gave it my shot, but I'm going to walk away if this doesn't work. But every time I sort of gave myself that binary, it always worked out. And it's like a bittersweet feeling. It's like, oh, fantastic, I can keep moving forwards. But then there's this paranoia that further down the line, it's going to be super damaging. And, I basically set myself back by the period of time I've been working on this. So, lots of warning labels here. I'm more risk prone than most. Maybe because, I've got a dad who was an entrepreneur. It just seemed normal to me. So you've got to judge this carefully. And I don't know, maybe, if you're interested in aging and you're actually in a position of safety as far as like steady job and stuff, like try to sort of build from a position of safety. Don't take any risks, like especially on behalf of others if you've got little ones. If basically just take a measured risk. So yeah, if you've got something safe and steady, just use that as a way to work. You leverage that to basically work on this, which is where you want to go in the future. And secondly, I'd characterize this field slightly differently from, say, a normal biotech or a normal industry. I think it's half biotech and successful commercialization, all that stuff, but I think it's half an expedition. Quite like the Everest expedition. It's like a new frontier. Everybody's trying to summit this. Everybody's trying to climb this summit, and there's a lot of deaths along the way, but you find out new information. Everybody's really trying to get to the top of it, and the view from the top is just it's so exciting, right? The reality. Just imagine you wake up in the morning and you can decide to reset your age to something like mid-20s. It's just astonishing, right? And the reality is, what would you do with that extra 30 years, right? I tend to think the people that are going to do this, they're people that are going to love life, right? And they want an extra 30 years to do something particular. So, for instance, I like flying drones. So I sort of joke with my drone flying friends. It's like, I'm doing this day job in longevity, so I can just fly drones more. I could fly drones for an extra 30 years. So, you know, for me, like there's a real excitement on the other end as well, which is tangible. Obviously, I work on it a lot, but I think, you know, the reality is really exciting. I think people should be feeling that by now.
Well said. Yeah, totally true. Okay, Daniel, thank you so much for sharing your journey with us. It's just amazing what you've been able to do, and it was definitely not easy hearing, I guess, you sharing your experience with all the risk that you had to take and all the times that you came close to quitting. I think that's really important for people to hear, that it's, it's a tough journey for entrepreneurs, biotech founders, especially in longevity. So with that being said, let's just open it up for questions, and then I'll come back to you later, and we can close up the room with some rapid fire questions. But let's start taking some questions from the audience. You can raise your hand if you have any questions for Daniel. If not, I have a bunch of questions for him.
So I have a question. Daniel, very interesting story. Thanks for going into all that detail. Right at the end here, you mentioned that you came close to giving up many times. Can you share what was the closest that you came to really truly throwing in the towel, and what kept you from giving up?
Okay, so the first time, yeah, I mean, it sounds like it sounds like I'm, there's a certain degree of weakness, but it's just I think it's mostly looking at others, right? So there's no one else going through your journey, so it's looking at others is particularly troublesome as far as like testing yourself. So I'd done a load of bioinformatics on the computer, yeah, basically trying to predict what the cell line was doing. And I think the prediction was it was unfolded protein response. Yeah, so there's a lot of ways to activate this pathway. So we got a bunch of molecules together, and we basically started treating cells, and we got 0 the first time, right? And these are like 4-month experiments, like full time, and that includes the weekends, right? You have to go in and feed your cells at the weekend. They're like little babies, but you don't get any cuteness, and you don't get anything, right? They're just cells in a dish. So you have to keep these things going. Like I said, there's contamination. So somebody that doesn't care as much about their experiments will, they'll lapse, and there'll be sort of things floating in your cell culture. So that happened. That didn't present too much of a problem for me. I sort of soldiered through. But it was when I've been testing loads of things that activate this pathway, and I just got 0 and 0. And I think I just said that this last molecule, which was called 2-deoxyglucose, which is a glucose derivative, it was like one of the ones I hadn't tested. And I just said, look, like we were testing pooled populations, but there's variation within pooled populations. Yeah? So every cell in your dish isn't necessarily the same. There's always, it's quite often noted that different clones behave differently. So I basically cloned this cell line. So I was running like 10 times larger than I normally would. It was basically like do or die, right? This is either going to work or it's not. Basically I did the experiments. You collect all the samples, and there's this instrument called a pyrosequencer, and it gives out something called a pyrogram. So it actually looks like a heartbeat on one of those hospital monitors, and you can watch the signal come out in real time. And I knew exactly the shape that this signal needed to be, right, if I was gonna get something to work. And I don't know if you've ever had this feeling, but like sort of getting the adrenaline, and you can hear your heart, yeah. Like that's how much it mattered, because for me this was a life decision. And it was only one clone, right? It was only one clone in the first experiment, and a pooled population on the one clone and a separate pooled population. They moved to wild type, right? And it was just this amazing feeling. It's like, you sort of get shivers with music. But like I said, it's bittersweet. And I was like, oh God, I've got to spend another 3 years doing this, right? And then, how damaging is that going to be if, there's so many reasons these things won't become real world technologies, in clinical development, if there's any risks and stuff like that. So I think that was the most pivotal moment, that one there. I think there's sort of secondary moments where basically almost running out of money. It's like, do I take on even more money from people that I, have relationships with, basically risking their money, right? I might not be able to give it back, or do I press ahead? And I think, there was a decision to basically go ahead with some experiments. This was just before Jonathan showed up, and it was these Parkinson cells, right? Can we get rid of mitochondria in these bad mitochondria in these Parkinson cells? And it failed the first time again. And I think we only had enough money for the second experiment, and it worked the second time. Just really stupid reasons. There's so many things that go wrong in bioscience labs that are just stupid things, like the temperature was wrong, or the reagent went off because you left it an extra week, those sorts of things. But those were the two. But that moment at the pyro sequencer, that was the do or die moment for me.
Yeah, thanks. Quite a story.
Okay, are there any other questions in the room? No, if not, okay, then maybe I'll ask Daniel some questions. So right now, are you guys still doing CRO work, or have you expanded to a lab?
So initially we went just 100% with CROs. So there was a company called Total Scientific that's become our accelerator in Cambridge. So very high quality CRO. So not just taking orders, like really engaging in the experiments, making sure that you're not cutting corners, those sorts of things. So they were actually fantastic at the beginning. Just beyond the call of duty. For our animal experiments, we use the Jackson Laboratory in Bar Harbor, Maine. They just opened a CRO facility just when we needed one, and they were fantastic as well. But they were basically, that was we were one of their first projects, but they did a great job. But with CROs, because it's because you're the one that cares the most about your experiments. You're gonna put in the extra 10%. You're gonna go 110% to sort of do all the checks. A CRO is never gonna be, that interested. I'm not saying CROs aren't good or anything, but there's always that last 10% that they're not going to do, right, because they've got lots of different people. And so I was advised by Simon Westbrook that you should always go visit your CROs, meet them in person, because if you're just the other end of an email, you're not really, there isn't that human connection. If you can sort of build a relationship, and then you can then progress from that to emails, but they know there's a human on the other side, that tends to be much more successful. And it was the case. So we just had a great interaction by sort of following this. We then won a competition. So this was a startup accelerator called Accelerate Babram. So the Babram Research Campus is a great life science campus outside Cambridge. I think it's one of the biggest biotech clusters around Cambridge. And we got a check for £10,000. And we also got our own space for the first time. So we were able to do our own experiments. We didn't actually do very much during the accelerator period. But having the facility was great, and we could sort of package compound and send it to collaborators and things like that. And more recently with COVID, basically just labs became much more restricted. We so happened to basically do a lot of we sort of shifted, forgive the pun, to more bioinformatic experiments. It was basically around clocks and what you can do with the public data and stuff like that. And so we sort of took a period that was separate, not in the lab anymore. But more recently, we've just got hold of some lab space, and there's a all hands on deck experiment that's very similar to Shinya Yamanaka and the Yamanaka factors. So it's like an aging version of what Shinya Yamanaka did. So this is an all hands on deck experiment. I think I might even be brought into the lab because it's such a big bottleneck. Like if we get this experiment done, everything's good right afterwards, like on different fronts. I'm not saying like we're better or worse than anybody else, but if we can do this one thing and do it well, it's just going to be great. So I think we're all going to go into the lab for a period. Hopefully I can keep everything else on top of everything else in the meantime. But we're actually going to be in the Milner Institute. So my investor Jonathan has got an institute called the Milner Institute. So I'm very lucky to be there, but obviously, I think we had a door opened for us there.
Yeah, great. Maybe we can ask a couple questions about getting investment. So
Yeah,
The first time you
just a
went
second.
to
Sorry to interrupt here. We are very close to the top of the hour. So, Daniel, do you have time for more? I mean, other people, the people in the room might not have time. So, just wondering how much we're going to go over here.
Yeah, Robert, I've got as much time as you need from me.
Okay, so Nathan, maybe we can just remind people now. So this is we're at the top of the hour. I guess the lightning round questions, that kind of stuff, we'll do later. We are doing this again next week also, where we will have Mark Berniger and colleagues from Maximon in Switzerland talking about their work to try to help create longevity companies like Daniel's here. And just wanted to thank everyone in the room. If you have other commitments after, thanks for joining us. You can follow us on the app here on Twitter. You can also follow the Longevity Show account on Twitter and the website, longevitybiotechshow.com, and Nathan's newsletter, longevitymarketcap.com, for further updates. I think I will leave the stream running and the recording going at least a few more minutes. What do you think?
Yeah, totally.
All right. Okay, carry on.
Awesome. Maybe I'll just get one question in, and then we can get to David's question in the audience. So Daniel, when you were pitching your investor, how Jonathan Milner, how did that go like the first time? Because he, as you said before, was not really like an aging or a longevity investor to begin with. So maybe you can share some tips about to other founders who might be pitching non-traditional, and what I mean is non-longevity investors.
Yeah, sure. So it was more than that. So Jonathan had a policy which was no therapeutics. Yeah, so he invested in things that were more platform technologies. Therapeutics are very expensive, very risky, and they tend to need a lot more money than sort of angel investment level. So I basically just taught him scientist to scientist at the beginning. And it was just a scientific conversation to begin with. And then, yeah, like I said, he said, look, very interesting. Why don't you do a bit more work? And then it was sort of just a conversation from that point onwards. And we were talking to other people at the time, but it was mostly this conversation with Jonathan. And I think he made a few allowances. I don't know. Maybe there's something that he saw in me. I don't know, right? But I certainly wasn't a polished pitcher. I wasn't, polished in any way, to be honest. I just knew that I wanted to do this, and I was determined to do so. So I think if that can come through, I think, if you're an angel and you can see somebody that's just trying to do, like they're trying to crack the problem from every angle and going beyond the call of duty, it doesn't matter if the person's polished or not. You've got someone who you can work with. But then, yeah, since then, obviously, there's a lot you learn, right? Just talking to people and what works and what doesn't work. So I'm by no means a finished product at the moment, but I've definitely come a long way in my own opinion.
Yeah, great. Let's get to some audience questions here. David, what's your question?
Hi, thanks. Hi, Daniel. Daniel and I know each other from before. These days, we don't get to meet each other anymore, so it's nice to say hello. My question was really about because I think it relates to what you were saying about investors, and I think this is a particular thing for UK investors. I think UK investors are kind of wary of drug development because there's no revenue coming in, and it's all about developing IP and a long-term payoff, and that's kind of not what many UK investors are ready for. I just wanted to kind of ask you about your IP strategy and how you kind of get over those problems. And I'm sorry if I missed the first half of this, so you might have already said this already, but just if good to hear your thoughts on it.
Thanks, David. Yeah, good to hear from you. And I remember us having a chat some time back. Yeah, good to have you on the call. So yeah, IP. I touched upon this a little bit earlier. So basically, I just filed IP like it was something I did every day. With no prior experience, I just filed a USPTO patent. But then I quickly found out that there was prior art that made it, things that I was claiming obvious. So then I had to go back to the drawing board. We decided to involve people. So that was the first thing. And you need some IP, right? If you want to go the commercial route, IP is just necessary, unless there's always exceptions, right? But in the most part, that's necessary. And yeah, what I touched on earlier was, IP serves a few purposes. I think naively you think that it's to defend against people trying to copy you or rip you off, but actually it's more about raising a question mark, so that people that it costs them less to remove that question mark by interacting with you than it does to try and go around you. So that's sort of how you can build value. And then
but isn't really, sorry Daniel, it's basically your product, isn't it? IP in a way. If you have
that's correct.
If you're not getting revenue from anything else, then it's the IP that's your product's the way I think about it. And
correct.
And I suppose the question is, how do you best develop that in a way that attracts investors to think, okay, this IP is worth putting, you're worth investing in to get better value out of your IP.
Yeah, so firstly, I'm not the expert, and there's plenty of experts around Cambridge, like IP specialists. Yeah, so they usually have a specific expertise that's relatively close to what you're doing. They can basically provide the translation. Yeah, look, this is what you're doing, and this is how we would protect it, and this is what might be useful. They don't necessarily know everything about the investment front. That's for you to just get feedback from the real world about what people find valuable and what they don't find valuable. But yeah, you take their advice. You talk to lots of investors about what they want to see. Just try and sort of walk the fine line. Also on the costs I mentioned earlier, you can do this file and refile strategy. Withdraw and refile strategy. So you file a patent, and in the first year there's no details that are published. And if you 1 year down the line, and nothing's changed, right, as far as your tiny part of the scientific world is concerned, you can quietly withdraw that patent and refile, and you've still got priority over anybody else, but you don't run these escalating patent costs. So we've been able to keep costs relatively low that way. I don't think we're going to be able to do that from now on, but historically that's what we did.
Thanks, Daniel.
You're welcome.
Okay, next question, Carmen.
Hello. Thank you, Daniel. This was very interesting. I was wondering, you described in many ways how you've interacted in the kind of greater Cambridge area with labs, with incubators, with fellow scientists. And I was wondering whether you have found more generally the Cambridge community or Cambridge area, or Britain more generally, receptive to the kind of work you were doing, both amongst scientists for the support that they could give you or deny you in longevity-related research, and also amongst investors, insofar, of course, as you are able to compare.
This is a really good question, Carmen. So I've got a good story to tell here. So Cambridge, UK, it's sort of it has this romance, right? It's like Isaac Newton was in Cambridge, Stephen Hawking from Cambridge. It's really a very purist place. It's like science above all else. It's you lock yourself in a cave for 4 months, come up with that mathematical equation, and you will be celebrated. It's that sort of place. Now there is an entrepreneurial spirit as well, but it's really very science dominated. Yeah, it's like that is what's celebrated. And I sort of there was a point in sort of the history of Shift where I sort of got frustrated by the response from people just in the Cambridge area. So Cambridge is a very small place, but exciting place, but a small place. And There was always this itching feeling, right? If you're really trying to do something, where's the best place, full stop, right? Not the best place near your house, yeah? Where's the best place, full stop? And, you know, the west coast of the States, as far as density of, you know, longevity investors and interest is concerned, is unsurpassed. So there's Aubrey de Grey, for instance. He set up in Cambridge to begin with, but he moved out to the west coast, and he's in Mountain View now. And so I sort of basically went on the same westward journey. I think I said to my wife, I was like, look, I wanted to raise money to do a clinical trial, yeah, and it just wasn't happening in the UK. The risk level was too high. There wasn't a VC equivalent of Jonathan, yeah, and Jonathan just couldn't do this on his own budget. So we were trying to find somebody that was like the American version of Jonathan. So to Jonathan's credit, he basically said, okay, let's do it, right? Let's just go out there. And I was out there 3 months, and I took my wife with me. And you know, we packed up our whole house and put it into storage. So we basically didn't have a house anymore. We just had all our stuff in a storage container, because we knew that it was going to cost a lot to rent on the West Coast, quite an expensive place. We didn't realize how expensive. We ended up just in like very tiny accommodation. But yeah, I basically went out there to try and get the big money, and naively, I just thought, it's America, everything's bigger. I'll just ask for more. You know, just put a zero on the end of what I was asking for in the UK, and that'll be enough. And I think what was it? I went to Indie Bio, and there's a guy called Arvind Gupta, and I just asked him for 25 million, right? And we were nowhere near sort of the level that, you know, we would receive 25 million. But I just said, look, we need a clinical trial. I just need 25 million. And he just walked out the room, and everybody else was apologetic. But then I basically realized that you can raise the money, right? But you've got to up your game to raise the money. There's plenty of competition. It's not just that there's more money there. It's not like more is more. So I found that out pretty quickly. You need to perform on many more levels when you're raising that sort of money in the States. Like I sort of had to dress better, to get my hair cut a bit. It seems like superficial, but I was treated differently, right? You know, before and after. And yeah, basically there was more investors willing to take bigger risks out there. But I wasn't in the position, right, to connect with those sums of money. So I actually spent a lot of time quite upset, because it was just, you know, I think the learning experience was so steep. And my wife was with me, and she was like, you know, she was coming, we're going up and down the West Coast in a car, and she was holding the sat nav for me. She was, you know, she was doing things like just trying to help me out on all fronts, and I was just getting no after no. And actually the best thing that came out of the whole trip was an X Prize event. So it was the X Prize for Longevity event, and I've been invited to this. And basically a bunch of us in the room. It was in Los Angeles, and one of the first questions was, you know, what do we need, right, to move this forward? What should the prize be? And somebody was like, we need an aging biomarker. And I just put my hand up and I said, look, Steve Horvath came up with the clock, like nobody else had, you know, put their hand up. I said, look, Steve Horvath's come up with this. And Steve Horvath was sitting in the room, and he said, oh, I'm actually here. And I was like, that's fantastic, Steve, you should take over. And then I basically got chatting with Steve about the mitochondria and his clock. Like, we'd seen there been movement of the clock with mitochondria, and he'd picked things up. And basically he was excited, and he linked us up with one of his collaborators in the UK called Ken Raj, he's just he's like a diamond in the dust, this guy, Ken Raj, fantastic individual. Maybe not an accurate representation, but he's like the Robin to, you know, if Steve Horvath's Batman, Ken is Robin. But yeah, fantastic guy. And just to basically start having a conversation with these people that have just been doing it a lot longer than you, much wiser than you, in my opinion. It just means you can be focusing on the right things at the right time. But that was the best thing out of the trip. So I went there to get lots of money for a clinical trial, but I came back with a much better plan moving forwards. And it turned out that the plan that we wanted to, you know, basically the better plan didn't require 25 million. It's actually something we can do still with relatively small sum, not, you know, it's not a small sum of money, but we could do this in the UK. It's sort of in the sweet spot of what we can do in the UK at the moment. When it does come to a clinical trial, it's just a size thing with the States. It's just there is a bigger investor market, and yeah, if you want to do trials, it's a better place to raise the money, unless you can de-risk to the point that, you know, UK VCs come on board. But the States will always be willing to take that bigger risk, in my experience.
Thank you. That's very interesting. Thank you.
You're welcome, Carmen.
Daniel, if I can ask, how many people were at that X Prize event?
I think there was about 30 of us in the room. So I mean, I think others were aware of Steve Horvath. So maybe I was just, you know, I was just super enthusiastic. You know, you always get that one individual that's just always putting their hand up, and everybody's rolling their eyes. So you know, I put my hand up, but there was, I think that Laura Deming was there. Peter Diamandis, you know, he was there. It was sort of, you had to pinch yourself, right? And I just didn't want to say anything stupid. That was my main priority. Like, you know, don't put your foot in it and then you've got to, you know, carry that for the rest of your life. So I just tried to sort of be good. Aubrey was there. Yeah, it was a fantastic event.
Yeah, so one of the things that I took away from your talk that seems really like an important theme in your journey was the importance of having mentors, right? So it seemed like you have all these people in your network helping you out. So for other people out there, like founders who don't have a network, who are just coming in, can you suggest, or maybe do you have some ideas for them how they can get mentors or people in their network who can, you know, be in their corner?
Yeah, I think there's some fantastic shortcuts. You know, that basically the network is something that was pre-built, and I was able to walk through a door that might have taken others, you know, I don't know, 5 years, right, to get to walk through that door. So I completely acknowledge I was lucky, but if you have a card and you don't play it right, you know, it's not about, you know, being proud of yourself and doing twice as much as the next person. We're trying to get something done. Yeah, let's just play all the cards we've got. If you had more cards than me, I would expect you to play them. These days, you've got some fantastic shortcuts. So I think Y Combinator, you know, basically it's, you know, you go in there, Y Combinator is basically plugged into everybody. They try to help to the extent that, you know, you want help. And I think that's actually a fantastic development. That I think basically Y Combinator didn't have a longevity strand when I set up, and I just didn't think, yeah, I had a network, so I didn't feel the need to go on that program. But if you don't have a network, yeah, like I think Y Combinator has a longevity strand. There's a Canadian version of Y Combinator called Creative Destruction Labs, which I participated in this. So it's like a decentralized Y Combinator, based around an academic paper where they try and distill the principles of Silicon Valley and put that into a program. And that's a fantastic program. You basically get thrown into a room. I think there's like, you know, 30, like the people in the room have to be able to invest money. So they just come thick and fast about, you know, why what you're doing is wrong, how you need to improve. And then you basically, part of the program, you take on 3 mentors for 3 months and you update them. And that's fantastic. More locally in the UK, there's a new, I call it the Cambridge Y Combinator. So it's called Start Codon. And it's a bioscience focused accelerator. So we've basically got a local option. So, you know, if I'd started this later, I would have almost inevitably gone on Start Codon. So I'd basically recommend doing that if you don't have a network. But if you do have a network, obviously just make the most of them. So, you know, I made the most of my friends and family, and then, you know, they introduced me to their networks. So if you're trying to do something and somebody, you know, you have a good relationship with somebody, of course they're going to try and help you. But if you know there's something you're missing, you might have to do some cold calls. You know, try and reach out directly to people. I think there's a Bristol program. I can't remember what the name is, but one of the parts of the program is get 100 meetings. Yeah, you just need to just get 100 meetings. Just try and reach out to people, get as many of those as possible. And there's actually been some success stories out of this Bristol program. Like there was a major acquisition. I think it was a diabetes type project. But that's absolutely necessary, getting 100 meetings, and it's, you know, especially for investors. Yeah, it's like you have to take 100 investors, and 10 actually get back to you, and only one of them closes, that sort of thing. So yeah, you've got to be able to have a thick skin.
Can I just say that program's iCure, and we did that for Magnitude Biosciences, and yeah, it's really cool. You get basically 20K you get a early career researcher gets £20,000 or £30,000 to travel the world in 2 months to talk to as many people as possible, just to basically test your value propositions and
Yeah,
where your end users are. So it was really good.
And I've actually met a couple of people that are basically funded to go on these world tours, talking to people. So he just came to talk to me, and it was just a fantastic chat, because he, you know, the guy didn't have any responsibilities apart from just talk to me and learn as much and like soak up as much as possible like a sponge. So I think that's a very forward-thinking program. I don't know if there's any equivalents that you know about, Nathan, but that's a really good one. I can sort of link that later.
I think it's based on a US program actually in the first place. So I'm not sure what that original program was called though.
Okay, yeah, we don't want to take credit. We don't want to take credit. We don't deserve it. Yeah.
So Daniel, that's very interesting. Funny you mentioned Creative Destruction Lab. Excuse me, I caught up with somebody over in the Vancouver location, and I wasn't aware that that they were operating quite in that way. So maybe I'll follow up with you afterwards. I have a related question to this. You're talking about mentorship networks and so on. What would you say to people that don't have entrepreneurs in their family? And probably the majority of people out there, you know, they come from, they went into research or they pursued some sort of academic routes, and they, you know, many of the people in their family around them are risk averse. Amongst their family friends would be discouraging them from taking chances and so on. And you yourself mentioned that if you can, you know, if you can do something that is a measured risk, and you're not putting, you know, you're not unnecessarily putting others at risk, that you should do that. So what would you say to those, like, that essentially I'm asking, like, how much of an influence was your father, and what would you say to those that don't have that kind of influence in your life?
Yeah, so I mean, my dad's a double-edged sword, because he's my dad, right? The outward impression is that he's holding my hand, and you know, I'm the young face, right? Which, you know, I think in Silicon Valley it's better to have a young face, and then, you know, he's basically holding my hand the whole time, but you know, he's a great guy, right? And he's you know, he's really sort of, we've managed this relationship very well. There's been very testing times, by the way, where it's like I have to say to him, you know, like, we've got if we can't reach an agreement here, right, we're gonna have to basically, you know, it bleeds over into the family relationship. Like, there's been some testing times, but that's been fine. Now, if you don't have somebody like that in the family, which is most families, I've actually got a very simple answer, which is go work for somebody, right, that's doing something exciting, because they're sort of taking the risk. I mean, as long as there's a job opening, but go work for that person and just learn as much as you can, basically on somebody else's risk. And you know, with startups, sometimes they work, sometimes they don't work, but you're gonna learn so much so fast that way. And I think a lot of the most interesting projects these days, you know, it's obviously, I'd say 50-50, half in academia, half in some of these companies. So just go work for a longevity company sooner rather than later. I think that's the best shortcut.
Yeah, and if you're looking for a job in the longevity industry, there is a website called longevitylist.com. So if you're interested, yeah, just check it out. There's all sorts of different jobs in the longevity industry. It's not all biology and pipetting stuff. There's tons of computational stuff, business development. So I encourage people who are interested to go check it out. Sorry, I'm going to pronounce this wrong. Evgeny.
Oh, hi guys. Hi Daniel. Thank you for your talk. Maybe I missed it. I think David already asked this question. Am I right that you discovered this substance you use to control copy number of mitochondrial DNA in the MRC during a postdoc, right?
Yeah, that's correct. So I mean, the discovery was basically here's a cell line called A549. How do we distill what it's doing? And we basically distilled it down to a pathway and a molecule. But basically, this pathway unfolded protein response. You can take a bunch of things that activate that, but not all of them do anything. Not all of them have an effect on the mitochondrial DNA. Only some of them. One of them being 2-deoxyglucose. And to be honest, apart from that information, we know nothing, right? It's a big black box. And so there's just, when you've got a giant black box, it's just dying out for additional research, what's the target, things like that. And, my original research was in rare mitochondrial diseases, and I wanted to go into aging. So, I went into Parkinson's and aging, things like that. We basically did a lot of work that was novel. And we came across things that were novel, and those are our discoveries. So there is this sort of fine line, like how close between your original project and where you're going, right? And does that raise a question mark with investors? But this is something we've been able to manage.
So I'd like to ask, when you discovered this substance, did you have any IP problems with the MRC? Because if it's discovered in MRC, they should have some right for the molecule or for the proteins involved. I don't know. How did it work?
Yeah, so the MRC, they have rights to the original molecule, but that's not what we're developing. We're developing something that's not obvious, right? Obviously there's this thing going on which is a distillation, but what we've done is not obvious, and that's our innovation. So that is ours, and that's something that we can protect ourselves.
Oh, great. So basically, you discovered the pathway in the MRC, and then you decided, oh, I will develop another inhibitor or whatever it is to affect the pathway in a way, right?
Yeah, that's right. So there's a big black box. We've been able to, it's like Battleship. Yeah, we've been able to strike the battleship, but we don't really know where the rest of the ship is. Yeah, so basically when there's a black box, it's ripe for discovery, ripe for novel findings. And so we just, for me, that was the next step, right? Let's try and reverse engineer this technology a bit more. So we have a therapeutic prospect, which is something we didn't have and the MRC didn't have.
I have another question is about what diseases are actually linked to mitochondrial copy number, DNA copy numbers.
Yeah, good question. So, the disease, which was the model system that I worked in, which was MELAS, it's not copy number. It's actually it's copies with a mutation versus wild type copies. So it's the relative proportions of those. And if you have more of the copy with the mutant, you suffer the disease. So MELAS is one example. There's other diseases. I think 3 or 4 others. I think NARP is another one. LHON. These are all acronyms that I forgot exactly what they mean. But so and then you've got diseases that are caused by nuclear defects in mitochondrial genes. So 99% of the genes in the that make up a mitochondria live in the nucleus, and that's a very different. It's a different genetic system, right? So the mitochondrial genetic system is thousands of copies, mass redundancy, not much repair, but you can degrade what's damaged and you can promote what's undamaged and replicate that. But nuclear defects are much more different, and they're therapeutically, it's they're more amenable to things like CRISPR and things like that. So the particular type of disease that I'm working on, which is it's called heteroplasmy, a mixture of mutant and wild type, it requires a very specific tool. And there's only there's a few diseases that operate like this, but there's lots of mitochondrial diseases that operate in different ways. But yeah, I mean, at Shift, it turns out that we've actually come full circle. So the mitochondrial therapeutic, which is, it's something that we've got and we want it to reach a conclusion, but it's not what's going to carry us to the most effective human therapeutic intervention in aging, but it will do something in MELAS. Yeah, so that's where it was. The underlying findings, they were born in MELAS, and so it makes sense that you'd go back to MELAS or things that are similar to MELAS. So that's where we see the that therapeutic sort of arriving. At some stage in sort of preclinical development. Yeah.
Thank you. And also, I'm just wondering, what's the effect of this mutation, DNA, mitochondrial DNA copies with mutation on the normal aging? So it's clear that they participate in particular diseases, but do you have any idea how this mutation, mutated copies, affect normal aging or normal functions of organs?
Yeah, we asked this experiment. We asked this question in a mouse experiment by using the epigenetic aging clock. So we basically treated these mutator mice that are it's basically an exacerbated version of mitochondrial disease, but you get a premature aging phenotype. So we treated these mice with our drug, and we showed, interestingly, that this is a two-part answer. So interestingly, the mice don't show accelerated aging according to the clock. So whatever is going on in the outside is an acceleration of the phenotype of aging, right? Not epigenetic aging. So that's the first thing. But our therapeutic seemed to slow down that phenotypic aging on the outside, but it also slowed down the clock. Now, we don't know if this has got anything to do with mitochondria. It might be because we're affecting pathways that have an overlap with caloric restriction, and that's why we're affecting the clock. So we're not sure exactly what's going on there. Another thing that I would say is that sort of mitochondria keep popping up, in unexpected places. So Tony Whisker with sort of young blood or young blood factors, he did a single cell transcriptome analysis of heterochronic parabiosis. So basically stitching up a young mouse to an old mouse, trying to figure out how it is that the old mouse gets rejuvenated. And when you do an enrichment on the pathways that are activated in the old mouse that basically becomes younger, they're mitochondrial pathways. Yeah, so it's like the mitochondria driving the rejuvenation. Whatever's going on in this young blood, it's manifesting functionally as mitochondria. That's very unexpected. I mean, it was nice for me to see. It's like, oh, great. I didn't do that work, but I just happened to be positioned well. But that's very interesting. Secondly, there's a very recent paper from Steve Horvath, Ken Raj, basically doing an unbiased clustering of methylation data from all the different mammals on the tissues. And it's interesting that I think it's lifespan and aging actually form two different clusters. And it looks like the lifespan, cluster looks much more like, some things that are going on the cytosol, and then the aging cluster looks more like things that are going on the nucleus, which is basically our cellular computer. So I think there is a place for mitochondria. We're just basically, it's early days mapping that landscape. And like I said, right, we might find out after a bunch of work that we were working on the right thing all along. But where we started, we just didn't have the confidence to keep moving forwards. There's just a timing issue here. We want to basically sweep the landscape now and make sure this is the best lever. Yeah.
Thank you.
So it's very interesting, just to follow on to Evgeny's questions, but also you just mentioned something, Daniel, which maybe I misheard, or maybe you can clarify. You said that you had some mouse models that were showing phenotypic aging without the aging of the clock. Is that right?
Yeah, that's correct. So, if you go back to the hallmarks of aging, I call it like base camp. Like, we use an Everest analogy. The hallmarks of aging is like base camp. It's a great framework. There's finite types of damage, and like a vehicle, yeah, you remove that damage and you maintain function. But nobody had, one way that you can interrogate those different hallmarks is, let's perturb senescent cells and see what the clock does, or let's perturb telomeres and see what the clock does, and let's perturb mitochondria. So I think at the time, senescent cells had been tested, telomeres have been tested. They didn't have any effect on the epigenetic aging clock. It was very interesting. This unbiased biomarker wasn't affected by these hallmarks. So it's not to say they're not involved in aging, it's just they're not involved in epigenetic aging. And sort of an open goal was, what about the mitochondria? Yeah, so what happens when we perturb the mitochondria? Do we see anything? So, you know, our perturbation is mitochondrial DNA mutations. So you look in this mouse, this mouse has premature aging, like, it looks old in multiple measures on the outside and inside, but according to the clock, it's exactly the age it should be. Yeah, it doesn't show any acceleration. So again, it doesn't mean that mitochondrial DNA mutations don't contribute to aging. It just means that it's not contributing to epigenetic aging, and we don't know, like, epigenetic aging might be 10% of all of aging, right? Or it might be 90%. We don't know. We just know it's a very useful tool, right? It's linearly correlated with aging, the best, right, out of anything. That's what we do know, but we just don't know how it fits into the bigger picture.
Right. So let me just get one additional clarification here before I make the comment also. So are these experiments in your lab, company, etc.? Like, these are your own experiments that you're describing, not ones that you've read about?
Yeah, correct. So this is, I mean, we presented these findings, right? So they're not sort of confidential or anything. We haven't published them just because we just need to choose carefully what we do and don't do. Publications tend to take a backseat in commercial ventures. It's more about IP, and publications can complicate IP. So, but we did find that there wasn't an acceleration of the clock. So we basically showed that our model didn't represent an acceleration of aging according to the epigenetic aging clock, which is about as close to the most important process. People really believe in epigenetic aging as a framework, but we weren't showing acceleration of that. Yeah.
All right. So one final clarification: do the premature aging mice die sooner than the non-premature aging mice?
Yes, correct. So these mice, they die at 13 to 14 months. But that's a lot of time for a startup. So we were actually drawn to the clock.
Okay, but what I'm trying to highlight here is, if this is the first time I'm hearing this, by the way, if what you are saying is correct, accurate, then it's suggesting that there is strong counter evidence to the idea that the epigenetic clocks actually have anything to do with the aging that matters in model organisms.
So I should just clarify. So I'm talking about mitochondrial DNA mutations. Now, there's mitochondrial DNA copy number, which is, you have a certain level of copies of this genome, and you can vary the copies up and down. So there's a master regulator called PGC1 alpha. It's basically like a little knob. You can turn it and turn up the number of power generating organelles you've got. You turn it down. Now there seems to be I don't know, there's a certain amount that I know that I don't know if it's published or not, but there is a relationship between mitochondria and the epigenetic aging clock. I can say that, but it's not a direct link between the mutations and that clock. It's more about different aspects of mitochondrial biology. It's a fascinating organelle, and it seems to interact with the clock in different ways. It's just a very early research area at the moment, but it's looking interesting.
Right, but I mean, this is quite interesting what you're saying. If there are models of mutations or whatever it is that's causing this, that causes them to prematurely age, and there is not, and you find that the epigenetic clock does not correctly reflect that accelerated aging, that, to me at least, sounds like a very interesting direction to pursue. And to what you're saying, a lot of people believe in the epigenetic clock. The word believe is an interesting choice here, because I think a lot of people in general are looking at the longevity space and they believe very strongly that it's going to work. And that is a belief. There's no hard evidence, right? So it's, if there's counter evidence to or if there are counter examples to the idea that the epigenetic clock is uniformly indicating the progress of aging in an organism, then that would seem to me like one of the most interesting findings in what you've described.
Yeah. So Robert, I think just two-part question, two-part answer. Sorry. So firstly, the clock remains an alien technology. Yeah. So there's a lot that's known about the clock, but it was trained using a machine learning algorithm. Yeah. So the algorithm picked up features in the aging time course that go up and down with age, and together you put that into a sort of linear correlate. But the story behind that is not being told. So Ken Raj is actually the individual that is made the most inroads as far as telling the story of the clock. What are the cogs in the clock, right, that are driving the movement of the hands? Yeah, so that's that's a lot of his pursuit. There's some really interesting things that sort of maybe don't reach discussion. So, for instance, the methylation changes in the clock over aging are in the region of 3%, and methylation is a binary event, so you're either methylated or you're not. Yeah, so a change of 3% indicates there's a subpopulation of cells that are undergoing epigenetic aging. It's not all the cells, it's some of them. And anyway, it's a whole fascinating area about, where is the ticking of the clock coming from? Is it, single cells? Is it subpopulation? Is it a cell ensemble phenomenon? There's a lot of story that hasn't been told yet. Similar to my therapeutic, it's a big black box. It's an alien technology that we arrived at through unbiased processes, and we're reverse engineering it, and the clock falls into that same category. But like I said earlier, the clock is a window into aging biology. The problem that we've got is making biological inferences from methylation, methylation of a certain site and what that means functionally. It's a little bit difficult, and so we're working on it's basically putting the biology first, right? So how do we make a clock where the biology is first? So when we find a clock that works, we can tell a lot more story. Yeah? So this is, we basically, this is where we're going, and that's what I'll say.
So in a word, it is still fair to be skeptical of conclusions that lean too heavily on the epigenetic clock as a marker for physiologic aging.
Yeah, the clock is a sensor, but we don't understand, the machinery of that sensor and what it represents. It's just it moves in the expected ways, right? I think the belief in the clock is because it slows down with caloric restriction, it speeds up with things that reduce lifespan. It seems to behave reliably, but people forgot the more interesting question, which is, what is the clock, right? Like, what does it represent? They're more focused on the utility. Like, I'm doing something, how can the clock help me? I'm doing, somebody else is doing something else. How can the clock help me? It's more of that mentality. But Ken Raj is really, he's sort of picked up, the investigation on what is the core of this alien technology.
Yeah, so this is interesting. I'm definitely going to follow up further on what you mentioned here with Ken Raj and others. Certainly, this is the first time I'm hearing something like this. It kind of reminds of, when you have a hammer, everything looks like a nail. So when you have an epigenetic clock, everything looks
exactly. Yeah. Exactly. Yeah.
I want to go on to a couple of points that Yevgeny made before maybe asking something else here. So you mentioned earlier, and Yevgeny also brought this up, that the MRC takes a very large ownership stake in any fruitful discoveries that you would have made there. So I think you said it was 90%.
Yeah, so I mean, maybe I was just being juvenile, throwing my toys out of the pram, but I think the effort it takes to take something from academic level to real world technology is so gargantuan, that to only be party to 10% of the upside for me just feels wrong. And there's huge variation in policy between institutions. So unlike the MRC, University of Cambridge, it has the opposite policy. So you can keep 90% of the upside, right? If you develop IP within a University of Cambridge laboratory, you can basically claim up to 90% of the upside, 10% going to. And this is basically revenue from IP or something like that. So that's far more. I don't know. I think it's far more forward thinking, and Cambridge's got many more success stories. So I think when you see a policy like that, you see success stories from a very different policy. You just have to think: is this really the right way to do things? Am I going to be stuck with this weight around my leg, or am I going to take a bit of risk myself, try and do things myself, spend my own money, and just keep moving this project forwards? Because it takes a person, right? These things don't happen on their own. It takes a person.
It takes a team to move technology. No, I really appreciate you bringing up that point. So I mean,
yeah, a team, Robert. Sorry, I don't want to take all the credit. We've got a great team at Shift. It's a small team, but it's a great team.
No worries. So I mean, this is a very important point for founders, universities, investors, policymakers. What, in your experience, observation, opinion, etc., would be a fair percentage for the university to claim?
That's a very it's very subjective, right? And I don't think you're in a position. I think until you've actually reached the success, right? It's almost you want to tally the contributions at the end. You just want everybody to sort of get behind you and tally the contributions like that. But I do think the University of Cambridge policy is more forward thinking, and more in line with some of the. I mean, you just look at the success stories, right? Like if the institutions are getting it right, there should be success stories. What are the policies of those institutions? I think Stanford does a really good job. I think with some of their IP licensing on, I think it was recombinant technology. Like they basically took a big picture view of it. It was like, look, we want to get, we obviously want to get upside from our science, economic upside, but we don't want to do that at the detriment of what is, startups are about as fragile as it gets. We don't want to tax these guys before they're able to be taxed.
Right. So it sounds like closer to the lion's share going to the investigator rather than the funder or the university. We have at least one other person here in the audience. Andrew, if you want to join in the conversation here, just raise your hand. I think it'll be interesting to have a discussion about this, because it's obviously a very big point of contention for anyone involved in commercializing anything. Okay, let's bring Andrew up here. I do have a more scientific, technical question afterwards, if there's time. And again, just to remind people, so we're approaching on 2 hours. We're keeping the conversation going, but this is being recorded. So if you do come on stage, your voice and image are recorded. So Andrew, what are your thoughts?
Well, this has been fascinating and great to hear your story, Daniel, of how it all started and where it's going. Yeah, so many, sorry, pardon me, so many interesting thoughts and questions. And Robert, yes, I thought it was amazing that you touched on this issue of the disconnect there with aging and the clock. And it's going to be tough to publish those results. People don't want to hear that. And so it's going to be interesting if that's sort of kept quiet, or, is the epigenetic clock going to be a context-specific clock? What are your thoughts about that, Daniel? Of getting that sort of word out, that it's maybe context specific.
So I think the clock has, I think the approach behind the clock is so compelling. So Steve Horvath didn't just arrive at the methylation clock. He went through every omic layer available to him, in the public databases, and it just so happens that he can, create a compound methylation signature that's best correlated. So he exhausted the data available and he arrived at this clock. So we can't just dismiss it out of hand, just because, I don't know, just because it doesn't, it doesn't fit everything, and it doesn't match phenotype all of the time. So there's something important going on with that clock, but it remains to be elucidated. Now, I think there's an interesting conversation, which is this phenotypic aging. Yeah. So, where's the line between phenotypic aging and lots of sources of aging? So there's, for instance, if we exacerbate mitochondrial DNA mutations, we get phenotypic aging. If we have a defect in DNA damage repair, we get phenotypic aging. Hutchinson-Gilford progeria, which is a mutant lamin, we get phenotypic aging. So it's like all of these different sources are getting integrated at the phenotypic level, and so there's something that's, I don't know where all of the different things plug in, but there's some sort of, it's like sort of tribute. It's like streams joining into rivers, and then finally you get to phenotype, and the phenotype is the ultimate integration of what's going on in everything in the cell. So ideally you want a phenotypic clock, so you want something as close to what you're concerned about, which is whatever phenotype you can think about, but you want a clock that operates at the level of phenotype. So you want to get as close to function as possible. There's actually a fascinating technology. I think Calico mentioned this first. So it's called, what's it called? It's called cell painting. Yeah. So you basically apply 7 or 8 dyes to the cell, and you can basically stain the organelles in different ways, and you can get organelle length, organelle proximity to other organelles. It's about 150 measures. But these are phenotypic measures, yeah, and they're incredibly data rich because they're integrating everything that's going on, below the surface. And you can basically create an aging biomarker that is, I think it's as accurate as the epigenetic aging clock, but it's at the phenotypic level. So if you just have to ask yourself, what is it that you really care about? Is it the aging phenotype? Is it the underlying cause of aging? It's almost like the Big Bang, right? Where did it all start and how do we fix that? Or am I just concerned about the end result and fixing that? You just need to be very careful about what you're interested in and why, and what ideally we want to fix what started the whole problem, and we want to fix the phenotype simultaneously. That would be ideal, but maybe there's compromises.
Yes, I am a firm believer in the idea of cell painting and sort of imaging and sort of phenotypic assays, and maybe there is an option out there, right? I mean, again, the clock is a biomarker. We're hoping it's the sort of way we can study aging in clinical trial. And maybe
yeah, but you must always ask. You almost test your assumptions, yeah. Like, what is the clock? Like, if we stop investigating that, we're going to end up in a very dangerous place, right? Where we're putting a lot of risk on results on the clock, without it qualifying that, right? So you've got to keep asking that question. That's why I really I'm a big fan of Ken Raj. He's sort of taken up the role of doing this, and he's got some really fascinating stuff, but it's really up to Ken to talk about that.
Yeah. And Robert, I guess you pulled me up here on the thought of academia and how they can enable
basically the question of what is a fair division of ownership of discoveries that are in the lab that are being commercialized. So should it be 10% of the university, 90%, 25, 50? Like, it depends, but the question is, like, what do you feel is fair, and what will actually get people moving in that direction?
Yeah, it's a tough one, but I think if you view it through the model we have is VC, where you can either have royalty payments. So you should enable the startup, as Daniel said. It's a fragile beast, and you need to enable it to flourish. And putting payment structures up front before there's been any investment or revenue or profit made is a deterrent. So it should really be small up front. I mean, at minimum, sort of cover the IP costs at the first opportunity. But they really should be allowed to flourish, and there should be royalty payments or at least milestone payments at later days, typical to more of a VC. And we see that MIT does a great job of that. I think they're very sophisticated in the way they approach it.
So Robert, I just add that there's so much variation in the commercialization models between institutions. It would be fantastic if somebody did a proper analysis, right? Say Cambridge University and companies that have originated, MIT, Harvard, right? And just try and basically link the policy to the success rate. I know there's Northern European countries where the founder just keeps everything, right? So the institution just doesn't have any hold on the IP whatsoever. So basically they can do science, but if they want to commercialize it, it's all on them. And I don't know what the success rate is of that institution, but it's, the variation is very large. And I don't know what the connection is, but there might be a paper out there, but if somebody did that, that would be fantastic.
Yeah, that's a really good idea. Which countries did you say the investigator keeps full ownership?
I think it's an oil-rich company. I think it's a country. I think it's Norway. I think it's Norway where they have this, but I might be wrong. It might be Sweden, but I'm pretty sure it's Norway.
All right. Well, that's something to look into for sure. I do have a technical question going back onto the mitochondria stuff. Unless someone else wants to ask something. So,
hi all.
Person on the welcome to the stage. What's your question?
Sorry, yes, I have a question just on the most recent discussion around payments to the university and that sort of thing. So I am studying. I'm in healthcare supply chain, and I study pharmaceutical supply chains at Ohio State University. And so I was just curious, my research is really at the intersection of pharma and public policy. And the policies that you're talking about, are those typically at the country level or are those at the university level?
Yeah, they're at the university level. So yeah, basically universities can set their policies, you know, how they basically commercialize research, and it's really up to them. I'm not aware of any top-down national initiatives, certainly not in the UK. Maybe that exists elsewhere, but from what I know, it's basically university specific.
Very cool. Thank you.
You're welcome.
So, Daniel, I have a question about the measurement of mitochondria on the one hand, and a separate question. I think this comment that you made about the epigenetic clock not lining up with premature aging models is very interesting. Like I said, it's the first time I've heard about this in the context, like myself, in a research context. I'm going to follow it up. But if you wanted to measure what's going on in the cell with the mitochondria without the epigenetic clock, what are the best available measures that you're using?
Yeah, so very simple answer. There's a whole company that was set up to do this. It's not the single cell level; it's just cell populations. But it's called Seahorse. So I think it's called the Seahorse Flux Analyzer. Sounds like something from Back to the Future, but it's just it's a machine that basically takes oxygen consumption measurements. So you basically treat the cells with various things that perturb energy production, and it can basically figure out how much oxygen your mitochondria consume, which is a proxy for function. And it's a very standardized assay. It's well commercialized. A little bit expensive to do the assay, but pretty accessible now.
So using that assay, can you detect the rejuvenation of the mitochondria independently of the epigenetic clock?
Yeah, that's correct. Yeah, so lots of people do this routinely. Like you can, I mean, before the clock, people used to look at mitochondrial function, and they looked at various different things, and yeah, mitochondrial function was one of them. So yeah, it's just an obvious thing to check if you're. I mean, I think a lot of the papers that that are published that use the clock, they always use alternative measures as well, right? So they don't put all of their eggs in one basket on the clock. So I think David Sinclair has measured, I don't know, autophagy and mitochondrial function. They always do those things in parallel, in addition to the clock, just to get out of this, this risk that, the clock is basically, it's a false positive, right? That's what you're worried about. The clock's saying one thing, but actually it's just the clock misbehaving. Yeah.
All right. My other question, sort of following up onto some of the stuff I think getting brought up. What is the maybe I should ask this the other way. So much research makes use of, including your own, as you described, mice, mouse models, and so forth. Are there any organisms that have, interesting properties with their mitochondria that would be, showing the same kinds of results that you're hoping to see here? And sort of related to this is, what is the smallest contained version of a mitochondria that exhibits the aging and rejuvenation that you are studying? So, for example, is it possible, I don't know the field, so I'm asking genuinely here, is it possible to isolate like a single mitochondrion or two, put it in some cell-like medium, in like a micropipette well or something, microwell, and just look at what's happening to it instead of like the entire cell or collection of cells?
Yeah, you can do that, Robert. And I think Nathan's done a great couple of articles on sort of mitochondrial companies and all the different things that are going on. There's some really interesting stuff. It's like, for instance, there's mitochondria just in your blood, like free living mitochondria swimming around, right? This is like a real finding, and it's completely nuts. So these things can actually sort of move around. I don't know what the circumstances are, but they're able to do this, and they were once free living organisms. So You can have a look at them. I don't, you see, I'm not a mitochondrial specialist anymore, right? That was actually quite a while ago now. So I don't really put all my energy and bandwidth on that field, but there's certainly some really interesting stuff. The more you get into mitochondria, it's like Narnia, yeah. It's like, oh my God, what's happening now? What is it? It's like the electron transport chain. It runs way above room temperature. I think it's like 50 degrees or something. This wasn't known before. So, this has real consequences as far as protein folding is concerned. And
so there are local temperature gradients inside the cell.
Yeah, like the engine is hot. I mean, it sort of makes sense after the fact, right? But they're very hot, and you've got to deal with that heat. And there's so much that hasn't been discovered about them. I'd encourage you to sort of read about it, but obviously don't become blinkered, because that's the risk, right?
No. Yeah. When you say blinkered, what do you mean?
I mean, it becomes so interesting that you forget there's research outside of mitochondria. Like, this is what I'm guilty of, right? It's just everything looks like a mitochondria, and you said this, like, everything looks like a nail when you've got a hammer.
Right. So I mean, no, I'm with you on this. I think people that are at least honest with themselves, who are working on biology, will sort of say, the more that is known about biology, or a particular area of biology, the more that you realize that you actually don't know. So now. Yeah. But the reason I brought up the mitochondrial model organisms and isolated models is, would it be helpful, do you think, in your work to have more advanced instrumentation and platforms of this nature, as opposed to being forced to work with only the existing assays, entire mice, etc.?
I wouldn't say that right now. I think the seahorse machine ticks most of the boxes. Yeah, it just gives you a really good idea of how the mitochondria are functioning, like in a basal state and under strain. Like maximal energy production and then basal energy production. So for me, that ticks a lot of boxes. There's not a huge amount extra that I'd need right now from that. So yeah, I think sort of technically, I think the field has got the tools at the moment. There might be some niche application that I'm not thinking about, but just coming back to, I think you're talking about organisms. So there's a really interesting finding in naked mole rats. So this was actually a George Church finding. I think it was way back, like 2009, and he basically did analysis of the naked mole rat transcriptome, to basically what, naked mole rat versus mouse. Yeah, a mouse doesn't live very long, and naked mole rat's about the same size but lives way longer, like 10 times longer. What's the biggest transcriptional difference, right, between those two, seemingly similar organisms? And transcriptionally, mitochondria are the most upregulated pathway in the naked mole rats. Yeah, it was like bang, it's just there. And George Church isn't a mitochondrial biologist, so that was very interesting to see that. It might have something to do with their environment. I think when it rains, the soil gets wet and they have to dig intensely, so their energy consumption just goes off the roof with the digging. So maybe they need extra mitochondria for that. But there's an interesting story there.
There's a lot of interesting directions to take. I didn't want to monopolize the conversation here. Maybe one last question for me, and I'll pass it on to someone else. You did say that you're not a mitochondrial biologist, like you're not focusing on that so much anymore, but for the science nerds in the room here, and I'm one of them, what are the best, say, 1 or 2 textbooks to get up to speed with what is currently known about mitochondrial biology?
Okay, so there's a great book with a great title by Nick Lane, who's an aging scientist at UCL in London. It's called Sex, Power and the Mitochondria, just because mitochondria are involved in those two things. And it's actually quite an old book now. I think he's done a follow-up to this book. But it's a great read, and it just takes you down the rabbit hole of mitochondria, and like basically, why do we have 2 sexes? It seems like the mitochondria are involved in that. Basically, the reason we can do what we can do as big multicellular organisms is that we internalize that energy production. So if we didn't sort of fold that membrane on the inside and have this huge surface area, we wouldn't be able to pump protons as much and have as much energy. It's really that event, the sort of eukaryotic cells, that's swallowing of an organism and making it into a mitochondria, that was a defining moment as far as big organisms are concerned. So this book is, it's fantastic. You could probably read that in a day or 2 just because it's a good read.
All right, so Nick Lane, thanks. I'll look that up. Nathan, any other questions? Anyone else comment? You always have good questions.
Yeah, I guess if nobody else has a question. So Daniel, when do you think you can start going to clinical trial? Like, do you have a roadmap in your mind of how to bring these sort of technologies to possibly, humans and therapeutics?
Yeah, so right now, we're in a position where basically we have a clock that's been able to give us really great biological inference. So it's not the most accurate clock under the sun. It hasn't been optimized for absolute accuracy, but it's comparable to the accuracy of epigenetic aging clocks. But it's made out of genes, yeah, and genes have a big functional story. Now we have this list of genes. A lot of them are just exciting in their own right. If we wanted to today, right, we could just take a risk on some of these genes, express them, and just see if they're sufficient to rejuvenate. But we want to really do this systematically. So the Yamanaka approach, where he basically whittled down 24 factors to a minimum set of 4 factors, we're basically doing that for these what are rejuvenation factor candidates. So that process, I think, is going to take us a year to 18 months, just whittle it down to the common set. We want to do in parallel a experiment making sure that we maintain fibroblast identity with whatever set of factors it turns out is the minimum set. Make sure that we're not running the risk of basically tipping pre-malignant cells into cancer, which is a risk at the moment with these reprogramming. So once we've got that minimum set, then to be honest, at that stage, I think it's it's basically you just run the preclinical clinical gauntlet. The bottleneck really is just setting up what are those therapeutic candidates, right? What are those genes first of all? Are we absolutely sure of those? Can we validate those? And then once we know what they are, there's different routes, right? You can go therapeutic mRNA, which is a relatively new approach, but there's so many people taking the Pfizer vaccine now. The safety is, there's this doors opened, right, that you don't have to worry about safety exists, so you can go that route. Or you can go the small molecule route of mRNAs are problematic from a delivery perspective, but we really just want to whittle it down to those core set of rejuvenation factors. I don't know what we're going to call them, but at the moment we've got these bioinformatic predictions. Let's test the real world. Yeah, let's go to the real world, test reality, and see if we can do it, right? So for us in the next 18 months, it's all hands on deck. We might, there might be zero. Yeah, our bioinformatic predictions might be out, and we might need to go back to the computer. But just from what we're looking at, it looks very promising. I think I did have a discussion previously. Steve Horvath in his most recent paper with the mammalian clock, unexpectedly, the mammalian clock, it says that neuron genes and neurogenesis, neuron development genes are downregulated with aging. And we're seeing the same sort of theme crop up in sort of rejuvenation time courses. So it's very nice to see an unexpected finding on his side, and we're seeing it on our side as well. So there's just a lot of reasons for us to be, optimistic about what we're seeing. It's like there's, some usual suspects show up, right? It doesn't prove that what we've got is correct, but it's just reassuring. Like the clock, the epigenetic clock, it slows down with caloric restriction. That gives us confidence in the clock, right? It's behaving as expected. Our gene list is not as expected, but there's a lot of things that give us confidence.
Yeah, thank you. Let's see, do we have a question from Monty?
Yeah, I came in late, but I definitely am really into doing anything that I can for the sake of longevity. I'm trying a number of different protocols to see, what I'm comfortable with, and just some of the things that I'm currently experimenting with. Let me see, include, for instance, fasting, caloric restriction, and there's a type of supplement called berberine. It's supposed to be similar to metformin, but for for whatever reason, don't want to go the route of metformin,
Yeah.
it's been recommended that, we consider berberine. And let's see, what else am I doing?
Yeah, Monty, sorry.
Go
We're
ahead.
discussing sort of the biotech scene here.
Oh, okay. I'm sorry.
No, I appreciate your
Right.
thoughts, but maybe we can come back to that another time.
Robert, I can give a short answer because I did some personal experimentation, right? Because it's for the cause, right? You just want to take risks. Now,
Go ahead.
so just quickly, so I was a big fan of Dale Bredesen, and I don't know if you're familiar, but Dale Bredesen worked at the Buck Institute, and he came up with basically a model of Alzheimer's that you could address therapeutically through changes in diet and supplements and things like that. And I really believed in this, and I thought this was going to be like a book like David Sinclair's Lifespan. It was called like Ending Alzheimer's. And basically, I figured out that you could, instead of taking all these expensive nutritional supplements to reverse Alzheimer's, because I wanted to do that as an insurance policy, you could get most of it in a pregnancy supplement. I think it's called Pregnacare Plus. So for a brief period, I decided to take pregnancy supplement. And there's, it's quite funny, right? Sort of my girlfriend's parents show up, and there's pregnancy supplement all over the house, and they're like, "What's going on?" And then my girlfriend has to explain that actually it's her husband's taken the pregnancy supplement. But this is the really interesting thing, Monty. My nails grew much better and my hair grew much better, right? So it wasn't like, I actually felt some benefits. And when I came off the pregnancy supplement, because it has too much iron, which can cause toxicity, so no longer than a month. When I came off it, my hair wasn't as good and my nails weren't as good anymore. So if you love your hair and your nails, I'd take Pregnacare Plus.
Okay, I'll keep that in mind. I'll just listen in to see if there's anything I can get out of whatever research that you guys are up to. Thanks.
Well, you just heard that maybe taking pregnancy supplements might actually help.
Yeah. Okay.
Please seek medical advice. I'm not a qualified sort of doctor or anything. I'm just a PhD doctor.
Caveat. Very important caveat. This is not medical advice. Do not do anything without consulting a professional, your doctor, etc. But thank you for the question. So Nathan, Daniel, we're going on past the 2-hour mark here. How would you like to proceed?
I mean, I'm here until everybody's done with me. Just because I'm actually on holiday now. So this is tomorrow's holiday. So I'll just keep going.
Maybe I can ask a general question for everyone here. So for people who are interested in starting companies in the longevity space, founders, maybe they've never started a company before and they're just starting out. There are some options that we discussed that Daniel talked about, different accelerators like Y Combinator, Creative Destruction Labs. There was a couple that you mentioned in the UK as well. But if you could create such an accelerator yourself, what would you focus on? Like, what would be the main sort of ideas or methods or structures in such an accelerator that you would?
Nathan, I can start if you want. So I mentioned there's a Bristol program where they basically pay you money to fly around the world talking to people. Yeah. So if you can find the meetings, you just get the lay of the land, right? Just figure out what's working, what's not working, what's the state of the art. No obligations outside of that, right? It's just a big research task. I think that's a fantastic initiative. I mean, I was able to do this not through an accelerator, but just through Jonathan, who's, I'm just very lucky to have Jonathan. But Jonathan sort of supported that by, allowing me to be out there and doing that. But I think, that would be a great first step in an accelerator, because, I think building the company comes after you've found something really interesting, you've formulated it, you've figured out how it might go wrong. The company bit sort of, builds a little bit later, but you really need to find the lay of the land first, yeah. Like where you're going to work and who you need to work with, or maybe you just attach yourself to a company rather than start something new. It's not always the right solution to start something, start a new company. That's what I would say.
The accelerator thing you're talking about, the UK Bristol program. So they fly you fly around the world talking to investors. Is that correct?
Anybody. So anybody that you just send emails. You just, it could be a researcher, it could be an investor, just anybody that will have you, which is where the 100 meetings come from. You just have to cold call as many people as possible without a network. But they just pay you to do this, right? Just go talk to people and find things out. And if you get somewhere, then they'll continue to back you. If you don't get somewhere, then you've blown your 20K.
Wow, that's crazy. So they pay you 20K to talk to people for, you said, was it like 3 months or something like that?
Yeah, and I had one of these individuals approach me, and I was able to basically download a lot of stuff, and he said this is fantastic. And I think just setting up that situation, right, with that 20K, that's very forward thinking.
Yeah, and I guess that was before COVID or something. Well, now that there's COVID, everybody can do these meetings, online potentially, right? So maybe you don't need 20K to fly around the world. You just need 100 different, I guess, Zoom calls.
Yeah, also, I think these Clubhouse talks are quite good, and you're going to be doing this virtual, co-working, which I think is a really interesting idea. So I think maybe that'll tap into some of these things which the Bristol program is trying to get at in the real world.
Totally. Awesome. Does anybody else have any ideas about this, Andrew or Evgeny, from the academia side? Like, if you were starting a company and you were looking for like an accelerator or something like that to help you get some connections, what would you want to see in such an accelerator?
I guess it should be qualified investors, first of all, because it might be very difficult to explain to general investors what you want and if it's really worth to invest.
Yeah, so Evgeny, I'd just add that I think there are collectives now. So there's Longevity Investors Network. I think Avier Norris is one of the guys behind that. I think he's at lifespan.io. So there's this, I think he sort of basically created this like angel network. And I think longevity.technology is also becoming like a network hub for longevity specific investors. So these things are starting to happen now, and they didn't exist when I started out. So yeah, I mean, it's you're going to get some really good feedback from people that aren't just going to dismiss you because longevity is a craze or it's just hype.
Thank you very much, Daniel. I also have a question about the connection between epigenetics and mitochondria. So do you?
Okay, go ahead.
Yeah. Yes. So basically, it's about what do you know any particular pathways involved, or particularly about copy number and epigenetic log?
I just want to give Andrew the chance, because we're on a certain thread that Nathan started. Andrew, were you going to talk about basically
yeah. Thanks,
and then
Evgeny,
I'll get back to your question after Andrew's just
it's a very quick point following on from the VC. I mean, obviously Y Combinator I think has done a very good job, but the to get into that game is quite a high bar. And so I think there should be something which has a lower sort of entry point, and I think Clubhouse can form that. But I think there's a lot of people who are serious about starting things, but maybe don't have their idea flushed out. And having people like yourself, Nathan, Robert, to help guide, whether it's just to flush out ideas, I think would be a real help.
Yeah, Nathan, what I want you to do, and this is up to you, but it's like the Wall Street bets of longevity. Yeah? It's like you have this Reddit thread, and everybody's just piling on board. And if we get it wrong, we get it wrong together. But if we get it right, it's fantastic. And like I don't know if that's the way it's going to go, but I just like the idea of that.
Totally. Yeah, I've been thinking about installing something on longevity list, maybe some sort of way for people to collect together and sort of trade ideas. I also have something else that might be potentially big coming up in the next few weeks, but I can't really announce it until later. I'll talk to you in private.
Evgeny, so I'll just go back to your point about epigenetic clocks and mitochondria. So, I mean, just to give an example with my compound, which I can talk about. So, I said that in mice, so we affect mitochondrial DNA mutations in mice, but those mutations aren't linked to the clock. So our compound's doing something, right? But it's not really through the mitochondria. And interestingly, when we treat keratinocytes, which are the sort of surface cells in your skin, if we treat those with our compound, we can actually make the keratinocytes live about twice as long, so that they keep doubling for twice as long. But it has absolutely no effect on the epigenetic clock. So these keratinocytes, their telomeres aren't, they're basically the same length they would be, when the control cells senesce. So there's something about this compound that's targeting mitochondrial DNA mutations that allows the lifespan of the cells to double. That has no effect on epigenetic aging. So the way that I divide this is that, there's the computer of the cell, which is the nucleus, which is sort of epigenetic aging, like, how well is your computer functioning? And then you've got the cytosol, which is basically homeostasis. And that really is what keeps the show going, right? Maintaining homeostasis. And that's more, that's, again, it's speculative, but that's, you can imagine that's more about lifespan, right? Maintaining homeostasis. It doesn't matter if your computer's malfunctioning, like the nucleus. As long as you're maintaining homeostasis, you can keep going. And I think there's examples where epigenetic age can just, if you have an immortal line, it just keeps going up and up and up and up. But the cells survive. So again, it's what are you interested in? Is it epigenetic aging and reversing that at all costs? Or are you interested in some phenotypes that aren't necessarily all about epigenetic aging?
No, I mean, I was just interested in general, because it's indeed unusual that these processes are uncoupled. But on the other hand, why not? Why basically epigenetic age should follow DNA copy number? I mean, mitochondrial DNA copy number. So it basically, it's especially if it's in just cell culture model.
Yeah, so Evgeny, there will be results published at some point in respect to mitochondrial DNA copy number and the epigenetic clock. But it's not for me to speak about that. So I'm aware of them, but I can't say anything about it.
Yeah, it would be very interesting to read. And in terms of accelerator, sorry, I'm just switching between topics. I think it also would be great, maybe just indeed combine academic people from academia and people with business experience, the way they do this in Entrepreneur First, and just focus on particular longevity field. I'm sure there will be lots of interest for this.
So following up to your point, Evgeny, what kind of business people are you looking for to get advice?
Well, I think there is in general, when people go to academia, they really focus, to become professor or something like that, and most of scientists don't really understand what's happening in business side, and basically not trained, and most of them never have a dream about business. So when you suddenly realize, okay, your postdoc is going to finish and you don't really want to be eternal postdoc, you might start thinking about business, but you don't have really understanding of business side. But on the other hand, there are people doing a business and interested in longevity, but have no idea about molecular basis and other stuff. So it would be great to connect these people in a way, and both sides could bring the value for the startup. So
I see. So you're thinking about like connecting a scientific co-founder with like a more business side co-founder and see.
Yes,
if we can connect
this is
time.
exactly what they do in Entrepreneur First, actually,
okay.
and it works in a way.
Yeah, that's a good point. There can be a good division of work between these two specialties. It's difficult to have both, I guess.
Because people like
Daniel
are rare, people like Daniel who understand biology and can at the same time run the company. It's rare qualities, right?
Yeah, I mean, Evgeny, what I would say is that I wouldn't underestimate the power of just getting out there and just talking to people. You're going to get a lot of no's, and it's going to be frustrating, but there are people out there that keep an open mind, and they do have a science background. But yeah, like you said, though, it is rare to have the business and the science side. I think scientists, they double down on what they do well, right? Build on your strengths, and a lot of the commercial stuff just isn't prioritized. But I think being mission led, it just forces you to learn what you need to learn when you need it. But those individuals are out there, yeah. So I think just keep talking. Yeah, it's not always going to arrive at your doorstep. So there's a bit of a responsibility to sort of try and find things which aren't necessarily well defined. Yeah.
Yeah, just to follow up that.
Sorry.
So
yeah, go on, Robert.
I think a very good point Evgeny brought up, that it's rare to find and same person. It's probably easier in some sense for someone with science background to go into the early stages of trying to get a venture off the ground, but then in the later stages, business is not easy. There's real skill involved in navigating negotiations and fundraising and communication and things like that. So Sonia Arison of the Foresight Health Extension Group and 100 Plus Capital has brought up this point that there's a lot of great ideas in labs, but it's hard to find people that can bridge the gap between science and the real world of business. I did want to ask also, like, you mentioned, Daniel, that just getting out there and talking to people and trying to work for others who have the experience that you're looking for is a very productive way of going about all this. What about from the business side of things? Like, suppose you have someone that is, and there's a lot of people out there like this, right? Who they have enough money to support ventures like yours, but they don't know how to find you or someone like you, or research projects in the lab that are promising, like the ones that you've pursued so far. So how would you suggest to them to proceed from that side?
So yeah, just to paraphrase Robert, so it's a business person that is trying to find the scientist, basically. Is that what you mean?
Yeah, essentially. Like, how do you source opportunities for investment?
So I think obviously I'm a fan of Nathan, so I think he's doing a good job of putting those two together. Like I said, there was these angel networks. There's some fantastic tools available now as well, though, like sort of shortcuts. Like there's David Sinclair's Lifespan book, which is a great. It's like inviting new people into the field. They could just read that book, and all of a sudden they're turned on to the field. There's many others beyond David Sinclair that have written these great sort of public consumption books. I think that does a lot of good. Beyond that, trying to find specific projects. I think it sort of boils down to the academic literature. I think if you're publishing as academic, you're more likely to, make yourself known if it's a good publication. So, it's a good question, Robert. I don't think I've got a clear answer to that. I think the closest of these.
No pressure. It's more, just something to think about. And if anyone does have ideas, or if you come up with an idea later, we can definitely come back to it. It's a problem that I think a lot of investors or people on the investment side or the people on the business side are struggling with?
Yeah, one thing I would say is that I think there's a really interesting role that sort of scientists, academic scientists have, and they don't realize how valuable this is. And it's like being a Sherpa. Yeah, so basically introducing this field, because Aubrey used to, this is what Aubrey's been doing forever. Yeah, but there's only so many meetings Aubrey can take. So there's this huge opportunity just to take somebody that doesn't know about this and bring it to their attention. There's just so many success stories now, like real world results, like the TRIM trial. Greg Fahey and Bobby Brook, they've actually reversed epigenetic aging, like in real humans, and they didn't do it dangerously. It was relatively safe. Like that, that's just it's not a pie in the sky, right? You can point to that. Just inviting someone into the field and showing it to them is just so exhilarating for that person, yeah. And then you become this Sherpa, and then naturally after you've done them a favor, right, which is bring this thing to their attention, they'll say, okay, so what's interesting and what are you working on? So I think these people are all over the place. So Jonathan was an example, right? It just happens, this was a while back. But there's people just waiting to learn about this, but just haven't heard because they've been preoccupied on, I don't know, other industries or things like that. So yeah, if you can find one of those people that is like basically virgin, right, to the aging field, and you invite them in, wonderful things can happen. And sort of more recently, we talked to some guys. I'm not able to talk about the details, but these are really big, bioscience entrepreneurs in the States, like the very best and the very best Silicon Valley backing, and they weren't aware of what was going on. And I was just introduced. I've got a friend who's basically linked up to these guys, and they've just piled in so hard. It's unbelievable. It's like they're just moving mountains, right, to get involved, and they're like changing their own plans and things like that. And these were just these were virgins to the aging field, and it just took somebody like a Sherpa to just lead them to the view. And I think that's a huge opportunity that's available to a lot of us.
So it's very interesting. Thanks for that input. I guess it's something that we should consider doing more of, also to try to reach out to people. I guess, like you're saying, with the example of Jonathan, I think it's Milner, right?
Yeah, that's correct. Yeah, Jonathan Milner.
I was actually I made it almost all the way through his CSAR presentation that you pointed me to before the call here. So yeah, he does seem very enthusiastic and is talking about a lot of the key activity in this field.
Yeah, he's a great guy.
So we are now 2.5 hours in. We are still recording this for anyone on the call. Nathan, Daniel, I may have to step out a little bit, but feel free to continue if you would like. I'm going to leave the Longevity Show account on to keep the thing recording, at least for a little bit. So yeah, how do you want to proceed?
Nathan, it's really up to you. I don't want you to hang on out of politeness, so I will wrap up if now's the best time.
I can stick around, but I think at 7 I have another call. But yeah, I'd love to keep talking if anybody wants to keep talking.
Right, so it sounds like we can go till 7 local time here in Toronto. Daniel, it's very generous of you to stay. I guess it's what is it, +4 or 5 now to the UK? So it must be 11:30 or 10:30.
It's fine. This is actually like a treat for me to talk about these things. So don't worry about it. Okay,
no. Always love to have you here. Of course, the conversations that we bring up here are very good, and this really went in depth into a lot of the stuff that I think is on people's minds. I did want to follow up on an earlier question I brought up, where I was asking about the closest calls that you had, where you were closest to giving up. You did mention the contexts, but I guess what I was really trying to get at here is what kept you going. What is it that when anyone that tries to do something even remotely like off the beaten path is going to encounter an enormous amount of setbacks, discouragement. trouble, Murphy's Law, that kind of stuff. So what kept you going? What kept you from quitting? What was your motivation and determination to keep you on the path, to keep pushing and keep trying, and even when it seems like there's almost no hope, keep trying another approach and that sort of thing?
Yeah, no, this is I wish I spent more time on this. So I think what leads you to giving up, right? And it's usually a comparison between you and somebody else, right? Comparison is like, I think there's a saying, right? It's like comparison is the source of all misery, or something like that. And it's, your friends are just moving forward with their lives whilst you're stuck on this crazy idea that you started ages ago. So it's like it's that delta between where you are and where everybody else is in their lives, and lots of things that you want, right, but you can't have because you've decided to do this thing. And then basically that forces a binary onto your the current situation. It's like, look, this needs to work out in a certain way if I'm gonna keep putting those things off. So I think what kept me going, right, was this. I mean, this is just, it's a funny thing. You try and search for something where you're always winning at something, right? And it can be minutely narrow, but the thing that always sort of propped me up, or sort of gave me a bit of extra spirit. People just ask, so what do you do, right? And I'd say, oh yeah, I'm sort of working in bioscience. They're like, oh, what sort of bioscience? And I said, well, we're looking at the aging process. And then just everybody's just very interested. It's very easy to people to plug into this topic. There's so many questions, the sort of layperson questions that are actually very interesting. And I just discuss about what I'm doing, and it's a good conversation starter. People are always very interested, and they'd always follow up, like, oh, so how's it going now? And it just seemed like I was the only person doing something interesting, right? I think maybe I'm being unfair to my friends, but nobody else was really excited, right, about what they were doing. It was mostly about their home lives, not their work lives, and I thought that was a real shame. Although my home life was suffering because of my work life, but I don't know. It just felt like I was doing something a bit, bigger, just sort of a bigger story, right? Something like that, and people were just interested in it, and that sort of kept me going.
So it was the excitement of the venture that or the direction that really kept you going, even through all the hard times.
Yeah, I think it's an adventure spirit. I don't know. I don't want to. There's other adventurers out there, but I just like that feeling that you're going into the unknown. You're throwing everything at it. Yeah, you're trying everything. It's just a very good state. If you're behaving like that, you're going to be finding new technologies. You're going to be talking to new people. It's quite a nice place to be, as long as you can sustain that, right, and you actually get somewhere in the end. But the actual feeling is very nice. So that sort of helps.
Nathan, I just realized we didn't do the lightning round questions or any of that stuff. I want to go with that, and then I have a question actually to put to Daniel, if there's time here. So yeah, I'll come back to that. You wanted to say something here?
Yeah, actually, I just wanted to comment on what Daniel was saying. I think that's interesting, like this idea of being like an explorer, right, on the frontier, and it reminds me of this biography I read, Endurance, and it's basically talking about the journey of Ernest Shackleton and his journey to expedition to Antarctica, right? And they encountered a lot of issues on that trip, and they almost all died, and there was just like this crazy sort of quest for survival. And I think that's very apt for the entrepreneur's kind of journey and the mission that they have, just like being able to endure through all this pain, and especially when you get rejected by all these investors. I thought that was such an interesting story that Daniel was able to provide, and it just really comes back to my mission, that I'm trying to grow a wave of passionate people who are building, funding, and championing technologies that extend healthy human lifespan, right? And if you know the word passion, right, you think it's, oh, you just really love it, right? But actually, the etymology of that word passion actually comes from Latin for pain, right? So like the Passion of the Christ is actually talking about pain, right? So it's this idea of trying to find people who are willing to risk and endure all the pain that comes with trying to do these missions to try and, you know, take science to the next level for the betterment of humankind. So I just really love, you know, the story that Daniel gave to us.
Yeah, I mean, Nathan, if you keep the reality, so you know where we're trying to go, right, which is the reality of resetting your biological age, you know, to mid-20s, right? If you keep that reality in mind, you know, it's like the light at the end of the tunnel. You can really throw yourself into the dirt. I remember, like, I did a lot of things to raise money before I got investment from Jonathan, but the whole time I was absolutely convinced I could do this, right? And the reality was so exciting. So, you know, I smashed up concrete. I did manual labor, just to tide myself over. I helped my brother, who basically does websites. He had a drone website that was, you know, sort of failing, and I helped prop that up, and that website actually got sold, and I reinvested some of that money into Shift. I just did all sorts of these things just as a side project, just to keep the train rolling forwards. But because, you know, you've got to keep that mission in mind and the reality that's so exciting in mind. And to be honest, if you you know, it's so exciting, right, that you don't mind it too much, right? You can sort of take the pain, but without that reality, you just feel the pain. You don't feel anything else.
I'll second that. If I can just add a counterbalancing thought here, which is, you know, it would be amazing if we can reset our biological age or gradually move back in that direction. However, it may also lead to very great disappointment if it turns out that it's more challenging than expected to do so. So, you know, you were talking about how something like comparison is the source of all misery or something. Well, another great source of misery for people, especially in the modern day, is expectations that are too out of line with reality. So just a thought there. But if I may add the other question I was going to bring up here. I ran an impromptu room on a Tuesday night a couple weeks back on Clubhouse here, and I put the title, "Do You Want to Live Forever?" And it actually caught quite a few fish with some very interesting conversation from a range of perspectives. And one of the things that, one of the themes that kept coming up in people's commentary and responses is that they don't have a clear vision of what an extended healthy life and, you know, long life into the future would actually look like. In fact, many people had expressed the interest in living a longer life in order to see what the future would look like, because they're curious. So the question I would put to you is, what is your vision of the future and what an extended life at you know, age reset to mid-20s would actually look like?
So this is something I want to do at Shift, and it's just to try and, you know, basically it's to try and make it clear, why are we doing this? Yeah. So there's obviously many different answers to this. One of them is, you know, just the health burden, right? Economically, this has to be done. It's like renewable energy. If we don't stop this inflating health burden, we're just going to be spending all our money on health and nothing else. So that there's obviously that, and there's a few other arguments. But for me personally, yeah, you know, I really enjoy being alive, yeah, and I enjoy new things and new technologies and just participating in what's going on. So for me, it's mostly about having more life, right? Because I like it. And so, like at Shift, what I'm going to encourage people to do is, like, what would you do with an extra 30 years? You know, would you learn 3 more degrees? Would you, you know, would you, I don't know, do some more droning, which is, I fly this drone with a headset, which is a lot of fun. You know, if I had unlimited time, I'd probably do a lot more of that. I think Nathan has this, he has this Lambo, was it longevity? Not Lambos, yeah. Honestly, if longevity was a reality, I would like to have a Lambo. I think I've earned it. And I'd like to drive that around Europe, yeah. Like, I'd do that with the extra 20 years. I'd drive around Europe in my Lamborghini. I imagine it's a reality, and Aubrey de Grey pulls up in a Lamborghini, yeah. That's pretty cool, you know, and he's earned it as well. So, you know, it's you know, what are you going to do with those extra years? So for me, having those extra years, I can do way more things, right? And, you know, you've got all this space travel and stuff that might happen. It's just infinitely exciting, right? We just don't have access to it at the moment. And we have a cap on what we enjoy doing at the moment. And there's just many reasons that I want to do this. You know, the obvious ones, but also some of these.
So in other words, just teasing Nathan here, what you're suggesting, Daniel, is not longevity, not Lambos, but longevity and Lambos.
Longevity before Lambo. So I said basically you need to earn it. Yeah, if you're going to do things that are indulgent, you need to do some really, you need to do a lot of good first, and then you can be a bit indulgent.
Fair enough. I can certainly side with that.
So Robert, if I wasn't in that room that you set up, are people not able to visualize what it will look like? So, for example, do we think the aging we do still age? We show signs of decline, but it's a different rate. Or are people still hung up on the fact that my birth certificate says I'm 150, but I look 20 years of age? What's the sort of that conversation around?
It was very interesting, Andrew. It was actually a whole range of answers, including the ones that you said, including also people that said they don't want to live longer, and they give various reasons for that. I think what I'm going to do, I'm very backed up on all kinds of stuff around the Q&A here. But what I will do is once we get the recording, today's recording actually will be much easier than the previous. We figured out using the second account here that's sitting in the audience here, Longevity Show, we're able to record this cleanly, and that will be a quick post probably to YouTube or something. But I'm going to try to make show notes of all the previous calls, including that impromptu one, because it was a very interesting set of responses. And some people were saying that, many people said, for example, that they wouldn't want to live longer unless their friends and family and loved ones were around them. So obviously, what's the point of living to 500 if you're not going to have the people that matter to you? Mm-hmm. But there was also, one interesting response I got from a woman that said that she didn't want to live longer was that she had seen a scene related to this. She had seen a scene in one of the Lord of the Rings movies of one of the elves who was immortal, but had wanted to marry a human. And her father had warned her, if you marry him, then think of all the time that you're going to be around after he dies by yourself. And then she had an image of herself walking in a forested area for like forever, basically, without the person that she was in love with in the movie. And I thought that was very interesting because here you have a work of fiction that is having a profound real world impact on someone here on Earth. So, I'm essentially asking in an open-ended way, like, what are the images, scenes, books, movies, what have you, or just imagination that you might have about the future? Myself, for instance, when I've thought about living not quite at the age of, say, mid-20s, as Daniel's saying, but say in the 30s, it would be the franchise of The Highlander, where it shows like a fictional world where some people are born, they live hundreds of years, they never die unless they're beheaded, etc. That shows a very interesting thing where it's like you can imagine yourself living through all these epochs of human civilization and all this kind of stuff, and it has that kind of excitement to it. But other people have other views. And so that the benefit of that room was to try to get a sense of the perspectives that are outside the typical perspectives expressed by longevity advocates and enthusiasts, because there tends to be kind of some groupthink in this community as well.
For sure. No, that's fascinating. I mean, I guess based on those things you were saying, I think we, no matter in our finite, relatively short space of life now, if we think of what we could achieve, we go through series of friends over our lifespan anyway. I'm not saying my friends are disposable, but there is going to be a inevitable stepping stone through that life.
Well, as it was put in the memorable graduation speech that was narrated by Baz Luhrmann, if you search for it on YouTube, "Wear Sunscreen," there's a good line in there that says, "Friends come and go, but a precious few you should hold on to."
Yeah.
And I just wanted to add one other thing, which is like, say this reality, which I think we're collectively excited about. What does that do to people's priorities, right? As individuals and as groups. So all of a sudden, you've got as much time as you need, right? You just there is no upper bound on the time that you need to address a problem. And so there's so many changes in the dynamics, right? What do we choose to focus on? How much money do we put into something before we declare it a success or a failure? But I think a lot of good things will start happening when you open up that time dimension, which is basically capped arbitrarily at the moment because we just haven't evolved beyond it.
That's a very interesting perspective. Good point that you brought up. I forgot about that one, actually. Another thing that people brought up in that impromptu room was that by having indefinite time, it would put less pressure on people to actually live life or to get things done. So there are positions on both sides of that debate.
Yeah, that's true. Absolutely, Robert.
Is that really true, though? Because I guess one experiment you could do is ask children who have not been informed about, death, whether they have, a strict sort of timeline on trying to do things or not. But that obviously is, not controlled because those are children. But I don't know. Can you guys remember a time before you knew about a death? I guess. Do you know what your ambitions were at that time?
I think we're definitely selected for survival. And so I think if you take away, what would be the selection pressure to remove our reason to run or to have motivation?
Yeah, no, I think I'm way out of my sort of, square of expertise, but yeah, sort of motivation. I mean, that's a whole area, right? It's like what motivates people. Why is it that jobs are good for you? I think that's a whole area in its own right. When I was very, very young, and I didn't know about dying, I think when dying is a very distant prospect, you do behave slightly differently. I can't remember exactly. I mean, when you're a kid, there's too many things that are different, right? So it's not like a controlled experiment, but I think you could just do that in your head. I haven't done that comprehensively, but I think the things I've mentioned already, just changing priorities and things like that.
Hi, everyone. I just wanted to chime in on this question. But before I chime in on the question, I have a question for everybody on stage, and it just needs to be one sentence so that I chime in after. I think, Daniel, you just answered why you want longevity, which is to experience things longer, correct?
Specifically, flying my drone, yeah.
Okay, Nathan, why you want to do longevity?
Well, mostly it's because I don't want people to suffer from biological aging. I just think these age-related diseases have to.
Robert?
I'm into life extension. I'm just not quite as diehard as all the rest of them.
Cameron?
Thank you, Laura. I think for me, it's for the sake of opportunities.
And opportunities, you mean having more opportunities in the future, I imagine.
Yes, a less finite horizon of opportunities for every one of us.
And you, Andrew?
Yeah, I think it's to stop age-related diseases.
And of all those things, why do you think would motivate people the most?
For people to jump on the train and get behind this, I think most people are intrinsically driven.
You mean for their own experience?
Yes.
I think so too, that basically we come into the world alone and we leave the world alone. It's like we can be part of a community and a society, but in the end, we live our lives independently as our own person. I do think that, however, with things like this, because they're so massive, it needs to have some sort of collective consciousness aim. When you think of going to space, what motivated people to be okay for the government to spend billions of dollars to achieve something that has nothing to do with them? It was because of a collective consciousness of people thinking, yeah, this is really progress. And in a way, I think longevity could be something like that if we can get people to really have a collective mind about it. But for it to be happen that way, I do think that talking about the things that matter to people is crucial. Life extension doesn't matter to people because most people don't even have enough money to live up to their 60s, never mind into 100. And I also think that focusing on issues that matter to people, if you touch them from their psyche and things that, rather than say, I want longevity because I would love, I mean, I think one of the key things is aging is really horrible. And we don't speak about it enough because we kind of like don't think about it, and younger people don't think about it that much. But we really need to almost bring how aging is to the forefront of what really happens there for people to think, oh, shoot, I don't want that future. Because nobody wants to end up like that. If they knew there was a choice, they would take it. And longevity is that choice.
I'd just comment, Laura, that I think there's a stick approach and there's a carrot approach. So the stick approach is scare everybody to death, right? You're going to get Alzheimer's and everybody's going to get something. But I think the Apple approach has been underemphasized. So, what are the great things about that extra 20 years?
It's not about making a stick approach of saying make people afraid of that, because I don't think making people afraid of things works either. No. But it's also about stopping talking about life extension or things that people can't relate to. And there's too much of that conversation in terms of when we talk about the technology itself.
I don't think there's any right or wrong answers here. I think there's personal answers, and I think when things become more serious, there's certainly collective discussions. I remember I had this this guy show up after my talk, and he'd realised that this science wasn't just make believe, and he said, "You can't do this." And I was like, "What do you mean you can't do this?" And he's like, "This is like an atomic weapon." And I was like, "It's not quite like an atomic weapon." I just was there until the very end with this guy, like an hour and a half, where he's basically trying to convince me not to develop these technologies because it's going to be the end of us, like atomic weapons was. So, yeah, there's a lot of opinions to consolidate, but I think I think we're up to the task, right, of doing that and having discussions and communicating and stuff.
I'm with you on that, Daniel. The carrot approach is probably emphasized, and people probably just don't believe that it's possible. They don't know what's going on in the research and all this kind of stuff. But if I can ask also, you just mentioned this debate that you were having with the guy who's comparing these technologies to atomic weapons. If I can ask, can you elaborate a little bit on what was discussed and who this person was or their background? Because that's a very interesting perspective that seems also quite rare.
So there's quite a few individuals in Cambridge that they focus on the scary sides, right? It's the unintended consequences, ethical considerations around, and this is a valid area, but sometimes it can interfere, right, with the very early technology and getting things up and running. And basically, he was just very concerned because he really bought into this, that this is a reality, that there was too much responsibility in too few hands. It was like I was making decisions on behalf of society, which I think is a little distant from the truth. There might be a part of, there's always a small truth to these things, but I just said to him, look, nothing's going to get decided in Shift Bioscience about your life. Okay, it's there's lots of checks and balances, and nothing's foolproof, but we're very early days, and I think basically the more people know about this, the more people can apply different valves and pressure points and make sure that it doesn't go in a scary direction. But I mean, we're just Aubrey articulates this very well. We're just trying to keep people healthy ultimately, and what's so dangerous about that? It's just an extension of medicine.
He articulates it now because for, I think, 10 years they were touting the life extension thing, and that wasn't working.
Well,
well, I mean, they said it on the call that you guys had, which I found it fascinating, and I think it's so correct. We can't speak to people telling them about life extension because that simply doesn't interest most people. And especially after COVID, there's a huge opportunity with people realizing that health is even more important than wealth. It's like the one thing that you just can't do without, and no matter how much money you have, you don't necessarily have health if you are not taking care of yourself, or you're exposed, might also show that money makes a big difference. And I do think that there is in the media a big push in saying things that longevity is, you find the articles from the New York Times and the opinion pieces, and longevity is very tied to billionaires right now, which is a dangerous game because there's a lot of animosity towards billionaires as it is.
All right. Yes. Sorry to cut you off. There is a perception problem. I think we hit upon some interesting stuff here at the end. I do want to remind everybody, we're now closing in on 3 hours, 7 p.m. So sorry to cut this short. Fascinating conversation. Daniel, would you be open to doing this again? Maybe we can continue where we're leaving.
Absolutely, Robert. And I saw that Mihai has joined. I'm going to set up a separate room, Mihai, and you can come talk to me.
Sure. Yeah, I just wanted to chip in a bit, but yeah, if it's late, that's grand. We can do it another time.
All right. This was fantastic. Thank you, everyone, for joining. Hopefully, this was informative, and we can do this again. We will do this again at some point. I'm going to close the rooms shortly and close the recording as well. Thanks for joining.
Yeah, thanks, Daniel, for sharing your story. It's just so inspirational. It makes me want to work even harder and endure even more pain.
Fantastic. And yeah, good to speak to you, Nathan. Have a good evening, Robert.
Nice to meet you, Dan. Nice to meet you, Daniel. Thank you.
You too, Andrew. Thank you. Bye-bye.
Have a nice evening. Bye.
Close it out.